In 2007, at the Primate Research Institute of Kyoto University in Japan, a chimpanzee named Ayumu sat in front of a touchscreen. Numbers appeared on the screen, 9 of them, scattered across different positions. The numbers were visible for a fraction of a second, then they disappeared, replaced by white squares. Ayumu had to touch the squares in the correct order, from the lowest number to the highest. Based only on what he had seen in that fraction of a second, Ayumu got it right, almost every time. Then they tested university students, young, healthy, intelligent human adults at one of the most selective universities in Japan. They trained on the same task, they practiced, and when they were tested against Ayumu under the same conditions, the chimpanzee beat them. Not occasionally, consistently. At the shortest exposure times, the gap between Ayumu's performance and the human performance was not close. Subscribe right now. Before this video goes any further, tap that button and subscribe. If this is the first time you are watching this channel, you are about to spend the next hour learning things about the animal closest to you on this planet that most people spend their entire lives not knowing. Subscribe now so the next video finds you automatically. This video is about what the Ayumu study revealed and about what it connects to across 60 years of chimpanzee cognition research, and about what the honest answer to the question it poses actually is. What would happen if a chimpanzee outsmarted you? The answer is more specific, more documented, and more illuminating about what human intelligence actually is than any simple comparison between the two species allows. The first thing to understand about the Ayumu result is what it does and does not show. It does not show that chimpanzees are smarter than humans in any general sense. The research literature on comparative cognition does not support any simple ranking of species by general intelligence. What it shows is that in a specific type of cognitive task, the rapid encoding and retention of the spatial positions of multiple items in short-term memory, the chimpanzee performs at a level that trained adult humans cannot match under equivalent conditions. The task is specific. The superiority is real and documented and replicated, and the reason for it is what makes the results so interesting. The human brain, in the course of its evolutionary development over the past several million years, underwent changes that produced the expanded cognitive capabilities that distinguish our species. Language, complex tool manufacture, cumulative cultural transmission, the specific kind of abstract reasoning that allows us to build models of things we have never seen and to communicate those models to other people across time. These capabilities required, among other things, a specific reorganization of how the brain allocates its processing resources. One of the things that appears to have been traded away in this reorganization is the specific type of rapid, comprehensive, spatially precise visual memory that the Ayumu study measures. The chimpanzee retained a version of this capability that the human lineage reduced in favor of the linguistic and abstract reasoning capabilities that our evolutionary history selected for. The term that researchers use for the chimpanzee's visual memory capability is eidetic-like memory, and the specific mechanism that underlies it, while not fully characterized, appears to involve a faster and more comprehensive initial encoding of visual information than the equivalent human system produces. When the numbers appear on the screen for 200 milliseconds, which is the blink of an eye, the chimpanzee's visual system captures and encodes the position of each number with a precision that the human visual system, dividing its attention differently and encoding at a different rate, does not replicate. The white squares that replace the numbers contain nothing that distinguishes them from each other. The chimpanzee touches them in the correct order because it is working from a retained visual snapshot of where the numbers were. The human is working from a partially encoded, rapidly degrading memory that the short exposure time has not allowed to consolidate fully. The implication for the question this video poses is immediate. If you were sitting next to Ayumu at that touch screen doing the same task, Ayumu would outperform you under the conditions where the exposure time is short enough to prevent the consolidation that human working memory requires. This is not a theoretical conclusion. It is an empirical one, produced by a study that tested this exact comparison and found this exact result. The chimpanzee outsmarted the human in a specific and measurable cognitive task under controlled conditions with replication. That is what would happen. The Ayumu result does not stand alone. It is part of a broader body of research on chimpanzee cognition that has accumulated across 60 years of field and laboratory study, and that has consistently produced results that revise the simple picture of human cognitive superiority that the common ancestor comparison might suggest. Some of these results are in the domain of social cognition, some are in the domain of physical reasoning, some are in the domain of learning and generalization, and some, like the Ayumu result, are in the domain of specific perceptual and memory capabilities, whose performance profile turns out to look different from what the human cognitive profile predicts. The social cognition of chimpanzees, which has been studied most extensively in the context of what researchers call theory of mind, the ability to model the mental states of other individuals and to use those models to predict and influence their behavior, produces results that are more nuanced than the simple claim that chimpanzees do or do not have theory of mind. The early research, which used the false belief task that developmental psychologists use to test theory of mind in human children, generally found that chimpanzees fail the task in the same ways that human children under 4 years old fail it. The conclusion that chimpanzees lack theory of mind was widely drawn. The subsequent research, which used tasks better designed to capture the specific ways that theory of mind would be expressed, in a species whose primary competitive challenge is not the linguistic coordination that human theory of mind evolved for, but the specific competitive social maneuvering of chimpanzee community life, produced a different and more complicated picture. The research at the Max Planck Institute for Evolutionary Anthropology by Joseph Call, Michael Tomasello and their colleagues demonstrated that chimpanzees understand something important about what other individuals can and cannot see, about what other individuals know and do not know, and about how other individuals will behave based on this knowledge. The specific experimental paradigms that revealed this understanding involved competitive food access scenarios where a subordinate chimpanzee could gain access to food only if it understood that a dominant individual could not see a specific food item. The subordinate chimpanzees that were tested showed a sophisticated understanding of the visual perspective of the dominant, seeking food in locations that were hidden from the dominant's view rather than in locations visible to the dominant. They were using a model of what the dominant could see, which is a foundational component of the theory of mind that the false belief task measures. What this research shows when it is placed alongside the Ayumu result and the broader body of chimpanzee cognition findings is an animal whose cognitive profile is specific, sophisticated, and in some dimensions genuinely comparable to or exceeding the human profile, while being substantially different in the specific dimensions that language and abstract reasoning occupy. The chimpanzee is not a simple or primitive cognitive system. It is a complex, specific cognitive system that was shaped by a different evolutionary history and that is optimized for a different set of problems than the human cognitive system. The specific problems that the chimpanzee cognitive system was optimized for are the problems of chimpanzee social life in the African forest, the complex competitive politics of a community of dozens of individuals, each with their own social relationships, alliances, and competitive interests, each pursuing their own reproductive agenda in a social environment where the outcomes of social interactions depend on who is watching, who is aligned with whom, and what the current state of the competitive hierarchy is. Managing this social landscape effectively requires the continuous tracking of individual identities, social relationships, behavioral states, and the specific competitive implications of current social events for the observer's own position and interests. The chimpanzee's social intelligence, which has been documented in extraordinary detail across decades of field research, is the intelligence of an animal that has been running this calculation every moment of its waking life across millions of years of evolutionary pressure. The human social intelligence that this channel's gorilla and chimpanzee series has described in the context of competitive hierarchy and territorial behavior is the same social intelligence that is being described here in the context of cognitive performance. The animal whose political sophistication Frans de Waal documented in Chimpanzee Politics, whose individual histories Goodall recorded across decades at Gombe, whose specific social strategies have been the subject of hundreds of peer-reviewed papers, is the same animal that beat the university students in the Ayumu task. The social intelligence and the perceptual memory are different capabilities in the same system, and the system is more capable, more specific, and more impressive than any simple comparison with the human would suggest. The language research on chimpanzees, which extends from the first attempts to teach spoken language in the 1930s through the sign language experiments of the 1960s and 1970s and into the contemporary research on symbolic communication and lexical comprehension, is one of the longest and most contested research programs in comparative psychology, and its results illuminate both the genuine communicative capabilities of chimpanzees and the specific boundaries of those capabilities relative to human language. The early attempts to teach chimpanzees to speak, which failed because the chimpanzee's vocal anatomy does not allow the precise articulation that human speech requires, established the limit of vocal language acquisition, but not the limit of the language capability that the chimpanzee's cognitive system could support. The sign language experiments that followed, beginning with the work of Alan and Beatrix Gardner with Washoe in 1966, demonstrated that chimpanzees could learn and use a vocabulary of manual signs to communicate with human trainers in ways that had structure and that were generative rather than simply memorized. Washoe learned over 100 signs. More importantly, she combined them in novel ways that she had not been taught, producing sign combinations like waterbird for swan and open food drink for refrigerator that went beyond the specific vocabulary she had been trained on. Whether these combinations constitute the kind of syntactically structured language that is specific to humans, or whether they represent a more limited associative generativity that stops short of full language, is a question that the research community has debated for 50 years without resolution. What is not in debate is that the cognitive system of the chimpanzee supports something that is meaningfully communicative, that is flexible and generative rather than purely reflexive, and that responds to training in ways that human language does not entirely predict. The chimpanzee that learns a sign vocabulary is doing something with that vocabulary that the simple associationist account does not fully explain, and the something it is doing is related to, but not identical to what a human child does when it acquires language. The relationship between chimpanzee symbolic communication and human language is one of the most actively studied questions in cognitive science, and the answers that have been produced are consistently more interesting and more nuanced than the simple yes or no question of whether chimpanzees have language would suggest. The tool use and tool manufacture of wild chimpanzees, which has been documented across multiple research sites, and which shows clear cultural transmission of specific techniques across generations within specific communities, represents a domain of cognitive capability that was, until Jane Goodall's initial reports from Gombe in the 1960s, believed to be exclusively human. The termite fishing behavior that Goodall described, in which chimpanzees modify grass stems and thin branches to create probes that can be inserted into termite mounds to extract the insects, requires the selection of appropriate raw materials, the modification of those materials through specific manufacturing steps, the understanding of the relationship between the tool's properties and the task it is used for, and the planning ahead behavior that brings tools to the termite mound rather than manufacturing them at the site. Each of these steps is a component of the tool manufacture and use capability that was used to define the boundary between human and non-human cognitive capability at the time that Goodall first described the behavior. The cultural variation in tool use between chimpanzee communities, which shows that specific techniques are learned within communities through observation and practice, and are not shared between communities that live in close geographic proximity, is evidence of the cultural transmission mechanism that makes chimpanzee tool use analogous in its social dimensions to the cultural transmission of technology in human populations. The chimpanzees of the Thai forest in Ivory Coast crack hard-shelled nuts using stone and wooden hammer and anvil tools. The chimpanzees of Gombe, whose range overlaps with nut-bearing trees and who have access to the same stones and wooden objects that the Thai chimpanzees use, do not crack nuts. The behavior is not the automatic expression of a cognitive capability in the presence of the relevant materials. It is a learned cultural practice, that exists in some communities and not in others, because it was acquired through the social learning process in some communities and not in others. This is the definition of cultural behavior, and the chimpanzee meets it. The planning ahead behavior that has been documented in wild chimpanzees, including the transportation of tools to sites where they will be needed, the selection of raw materials for future use before the specific need for those materials, has arisen, and the behavioral evidence from field studies suggesting that chimpanzees anticipate the locations of productive food sources and plan travel routes accordingly, represents a cognitive capability that developmental psychologists associate with the frontal lobe functions that develop late in human childhood and that distinguish mature human cognition from the cognition of younger children and of other primates. The documentation of these capabilities in chimpanzees has been controversial, partly because the alternative explanations that do not require planning ahead are difficult to rule out in field settings, and partly because the attribution of planning to an animal on the basis of behavioral evidence requires assumptions about the animal's mental states that the strict behaviorist tradition of comparative psychology is cautious about making. What the planning ahead research represents, whether or not the specific cognitive mechanisms involved are identical to what human planning involves, is another domain in which the simple account of chimpanzee cognitive limitation turns out to be more complicated than it first appeared. The animal that beats university students in spatial memory tasks, that demonstrates perspective-taking in competitive social contexts, that learns and uses symbolic communication flexibly, that manufactures and transmits tool technology culturally, and that shows behavioral evidence of future-oriented planning, is not a cognitive primitive. It is a specific cognitive system of considerable sophistication, whose specific capabilities and limitations reflect a specific evolutionary history in a specific ecological and social environment. The answer to the question this video posed, what would happen if a chimpanzee outsmarted you, depends entirely on the domain in which the comparison is made. In the specific domain of rapid spatial memory, for multiple items, which is what the Ayumu study measured, you would lose. You would lose clearly, consistently, and under conditions where additional training improves your performance but does not close the gap under the shortest exposure times. In the domain of language and abstract reasoning, you would win, not because the chimpanzee has no linguistic or abstract capability, but because the depth and the flexibility of human language and abstract thought exceeds what the chimpanzee cognitive system produces in these domains. In the domain of social intelligence, the comparison is more complicated, because both species have sophisticated social cognition, and because the specific cognitive tools that each species has for navigating its specific social world are adapted to the specific demands of that social world in ways that a direct comparison undervalues on both sides. What the Ayumu result, taken alongside the broader body of chimpanzee cognition research, actually reveals is something more interesting than a simple ranking. It reveals the specific shape of what the human cognitive revolution involved, the specific capabilities that were gained, the specific capabilities that were reduced or redirected, and the specific animal that is our closest cognitive relative and whose cognitive profile at a distance of 6 to 10 million years of separate evolution still overlaps with ours in ways that illuminate both what we share and what we have become. The chimpanzee that sits at the touchscreen and performs the task that the university student cannot match is not showing that it is smarter than us. It is showing that the human brain is not simply a more powerful version of the chimpanzee brain. It is a different version, pointing in a different direction, optimized for different problems, and carrying traces of the ancestral capability that its specific evolutionary history has partially redirected, but has not entirely erased. The numerical cognition research that complements the spatial memory findings from the Ayumu study has produced its own set of results that illuminate the chimpanzee's mathematical capabilities in ways that the popular account of animal counting does not adequately convey. Chimpanzees have been shown to understand numerical quantity in the abstract, to perform simple addition of quantities presented non-simultaneously, to understand the ordinal relationships between numerical quantities, and to select the larger of two quantities across a range of quantity sizes and presentation formats. These capabilities do not require the specific symbolic number system that human mathematics uses, but they represent a genuine understanding of numerical quantity as an abstract property of sets that is not simply a learned association between a symbol and a specific training stimulus. The mirror self-recognition test, which Gordon Gallup introduced in 1970 as a behavioral indicator of self-awareness, produces positive results in chimpanzees, bonobos, orangutans, and gorillas, and positive results in a small number of other species, including elephants, dolphins, and some corvids. The chimpanzee that recognizes its own reflection, using the reflection to investigate marks placed on its face that it cannot see directly, is demonstrating that it has a model of its own body appearance that it can use to interpret visual information about itself. This is a component of the self-awareness that is generally considered a prerequisite for the specific kind of theory of mind that the social cognition research described earlier requires, and the chimpanzee's positive performance on this test is consistent with the broader picture of social cognitive sophistication that the field research has documented. The empathy research on chimpanzees, which has been conducted both in field and laboratory settings, and which has documented consolation behavior, targeted helping, and the specific emotional responses to the distress of others that are the behavioral markers of empathy in humans, adds a dimension to the cognitive portrait that the strictly intellectual comparison between species tends to overlook. The chimpanzee that approaches a distressed companion after a conflict and provides the specific affiliative contact that reduces the companion's distress is doing something that requires, at minimum, the ability to register the companion's emotional state, to assess it as requiring a response, and to produce the specific response that addresses it. Whether this requires the same subjective experience of empathy that the human experience involves is a question that science cannot currently answer. What the behavioral evidence shows is that the functional equivalent of empathy, as defined by its behavioral outputs, is present in chimpanzee social behavior in ways that are consistent and documentable. Tonight, somewhere in the African forest, chimpanzees are sleeping in their nests, their eyes closed, their bodies resting from the day's activities. The specific cognitive architecture that produced Ayumu's performance in the touchscreen experiment, the rapid visual encoding that the human brain reduced in favor of language, is present in the sleeping animals ready for tomorrow's use in the social and foraging challenges that the forest will present. The community around them, whose competitive politics and whose alliance structures and whose individual histories their social cognition is continuously tracking, will be what it was yesterday when they wake. The specific cognitive tools they bring to tomorrow's challenges are the tools that millions of years of chimpanzee evolution has built for exactly those challenges. They are not our tools. They are not inferior tools. They are specific tools, built for specific challenges in a specific animal that we share this planet with, and that we are still, after 60 years of research, continuing to discover is more capable and more specific and more interesting than the previous generation's account of it was. Sleep well. The longitudinal research on individual chimpanzees at long-term field sites has produced something that the laboratory studies of cognitive capability cannot produce. The developmental portrait of a specific cognitive system across a lifetime. The chimpanzees at Gombe that Goodall has known since the 1960s, and the individuals in their community that have been born and raised and grown old under continuous observation, represent the most detailed record of individual non-human cognitive development across a lifespan that science has produced. What this record shows is that chimpanzee cognitive development follows a trajectory that shares structural features with human cognitive development, with the specific capabilities emerging in a specific sequence, and the social learning that transmits culturally specific knowledge occurring across an extended juvenile period that allows the accumulation of the experience that adult competence requires. The chimpanzee that learns to crack nuts in the Thai forest does not learn it quickly. It spends years watching its mother, attempting the task, failing, attempting again, gradually acquiring the specific motor skill and the specific knowledge of material properties that successful nut cracking requires. The learning trajectory is slow because the task is genuinely difficult, and because the knowledge required is specific and must be acquired through experience rather than through any innate program. The specific role of play in chimpanzee cognitive development, which has been studied both in wild and captive populations, illuminates the developmental function of the extended juvenile period that chimpanzees, like humans, have evolved. Play behavior in chimpanzees involves the practice of motor skills, the testing of social relationships, the exploration of the physical properties of objects, and the specific kind of playful problem-solving that engages the cognitive systems without the performance pressure of genuine need. The juvenile chimpanzee that plays with sticks and stones and the properties of water is doing something that is functionally analogous to what a human child does when it plays with blocks or sand or water. And the cognitive engagement that the play involves is part of the developmental process that builds the specific capabilities the adult will need. The extended juvenile period of both species, which postpones reproductive maturity in favor of extended learning and development, is one of the most striking parallels between the two evolutionary lineages, and one that reflects the importance of learned rather than innate capability in both species' ecological and social success. The specific research on chimpanzee numerical cognition that has been conducted at Kyoto, at Leipzig, and at research facilities in the United States and Europe has consistently found that chimpanzees can understand and work with numerical information in ways that exceed what the simple conditioning accounts of animal cognition predict. The research by Tetsuro Matsuzawa and his colleagues, which has been running at the Primate Research Institute in Kyoto since the 1980s, and which Ayumu's participation represents the continuation of, has documented a range of numerical and cognitive capabilities in chimpanzees that have progressively revised the understanding of what the chimpanzee cognitive system can support. The specific finding that chimpanzees can learn to use Arabic numerals to represent numerical quantities, to use these numerals in computational operations, and to transfer this learning to novel numerical situations, is evidence of the capacity for symbolic representation of abstract concepts that was previously thought to be either absent or severely limited in non-human primates. The research on chimpanzee innovation, which examines how novel behaviors originate in chimpanzee communities and how they spread through the social learning processes that the community provides, has documented cases that illuminate the specific cognitive process of behavioral invention in a non-human primate. The introduction of novel behaviors at research sites where provisioning or other human activities have created novel problems or opportunities, the behavioral responses of individual chimpanzees to novel objects or novel environmental challenges, and the longitudinal tracking of novel behaviors as they spread through a community via social learning, all provide windows into the cognitive process that underlies behavioral innovation. What this research shows is that novel behaviors originate in specific individuals whose cognitive flexibility and exploratory disposition leads them to produce behavioral responses to novel situations that other individuals observe and incorporate into their own behavioral repertoires. The process is recognizable as innovation and cultural transmission, and the cognitive mechanisms that drive it are the mechanisms of the flexible, experience-based learning system that the chimpanzee's cognitive architecture is built around. The comparative study of chimpanzee and human cognitive development in the first years of life, which has been conducted by researchers who apply identical test batteries to human infants and to chimpanzee infants at equivalent developmental stages, has produced some of the most directly informative data available on the specific cognitive similarities and differences between the two species. The results of these studies, particularly the large-scale comparative study conducted by Hermann and colleagues at the Max Planck Institute that tested hundreds of human children alongside dozens of chimpanzees and orangutans on a battery of cognitive tasks, showed a specific pattern. Human children and chimpanzees perform at comparable levels on the physical world tasks in the battery, the tasks that involve understanding the physical properties of objects, quantities, and spatial relationships. The human children substantially outperform the chimpanzees on the social world tasks, the tasks that involve understanding the intentions and knowledge states of other individuals and using communicative information from social partners. This specific pattern of similarity and difference is consistent with the broader picture of chimpanzee cognition that the research has built across decades. A cognitive system that is sophisticated and capable in the domains of physical world understanding, and that differs from the human system, most specifically in the social cognitive capabilities that language and the human form of cultural learning require. The brain organization of the chimpanzee, which has been studied through neuroimaging research that became possible as MRI technology was applied to great apes in controlled contexts, shows a pattern of structural similarity to the human brain that is expected given the shared evolutionary history, alongside specific organizational differences that reflect the divergent evolutionary trajectories of the two lineages. The areas of the chimpanzee brain that are involved in social cognition, including the regions that are homologous to the human prefrontal cortex and the superior temporal sulcus, are present and structurally similar to their human equivalents. The areas associated with language in the human brain, including Broca's area and Wernicke's area, have homologous structures in the chimpanzee brain that are active in contexts involving communication and social interaction, though their functional role in the chimpanzee is different from the role they play in language processing in humans. The structural similarity between the two brains is the neural expression of the genetic similarity between the two species, and the functional differences between the two brains reflect the specific directions that each lineage's evolutionary history has taken the shared ancestral neural architecture. The specific research program at the Yerkes National Primate Research Center in Atlanta, which has produced some of the most detailed laboratory data on chimpanzee cognitive and social behavior, over the past several decades has included studies of lexical comprehension using a custom symbol system called Yerkish, studies of social learning and behavioral innovation, studies of cooperative problem solving, and studies of the specific conditions under which chimpanzees share resources with others. The cooperative problem-solving research is particularly relevant to the question this video poses, because it reveals the specific conditions under which the chimpanzee's social intelligence is applied to joint problem-solving with partners. Chimpanzees can solve cooperative tasks that require simultaneous action by two individuals, but the specific conditions that facilitate cooperation, the specific social contexts that make cooperation more or less likely, differ from the conditions that facilitate human cooperation in ways that reflect the different social architectures of the two species. The chimpanzee's cooperation is more conditional and more sensitive to the competitive implications of the cooperative interaction than human cooperation typically is, which reflects the highly competitive social environment in which chimpanzee social intelligence evolved. The question of consciousness in chimpanzees, which is the deepest and most philosophically contested dimension of the comparative cognition research, is one that the scientific community approaches with appropriate caution, but that the behavioral and neurological evidence pushes inexorably toward. The mirror self-recognition, the empathy, the theory of mind evidence, the future-oriented planning, the individual-specific emotional responses to loss and to the deaths of community members that Goodall documented at Gombe. All of these behavioral phenomena are the kinds of phenomena that, in humans, are associated with subjective experience. Whether chimpanzees have subjective experience in the sense that philosophers mean when they use that term, whether there is something it is like to be a chimpanzee in the way that there is something it is like to be a human, is a question that current neuroscience and current cognitive science cannot definitively answer. What they can say is that the behavioral evidence does not support the exclusion of subjective experience from the list of possible explanations for what chimpanzees do and why they do it. The specific research on chimpanzee memory that extends beyond the spatial memory findings of the Ayumu study has documented impressive capabilities in other memory domains as well. The long-term memory of wild chimpanzees for the locations of food sources, for the individual identities of conspecifics they have not encountered in years, and for the specific social relationships and histories of individuals in their community, is a memory system that functions across decades of individual experience. The research at Gombe that has tracked specific individuals across more than 50 years of observation has documented cases of chimpanzees recognizing and responding to siblings they have not encountered in years, of mothers recognizing the infants of daughters who dispersed to other communities, and of males maintaining the specific competitive relationships with distant rivals that the patrolling and intergroup dynamics of chimpanzee community life require. This long-term social memory is the foundation of the political intelligence that Frans de Waal documented, because the alliance politics of a chimpanzee community depend on the accumulated history of relationships between specific individuals across years of shared experience. The research on deception in chimpanzees, which examines whether chimpanzees can intentionally produce false information in a social partner, is one of the most direct tests of the specific social cognitive capability that full theory of mind requires. The ability to model another individual's mental state and to intentionally create a false belief in that mental state. The laboratory studies of tactical deception in chimpanzees have produced mixed results, with some experiments producing clear evidence of behavior that can only be explained by the chimpanzee's understanding that its action will produce a false belief in the observer, and others finding alternative explanations that do not require the attribution of intentional deception. The field observations of what appear to be deceptive behaviors in wild chimpanzees, including the specific cases of food concealment and the misdirection of attention that field researchers have documented in competitive feeding contexts, are consistent with the capacity for intentional deception, but are difficult to analyze at the level of mechanism in field conditions. The research leaves the question of intentional deception in chimpanzees genuinely unresolved, which is itself informative. The behaviors that the question requires are present, the mechanisms that produce them are unclear, and the uncertainty is evidence of the genuine complexity of the cognitive system involved. The research on causal understanding in chimpanzees, which examines whether chimpanzees understand the causal relationships between events, rather than simply learning associative connections between them, has direct relevance to the question of what makes chimpanzee tool use the sophisticated behavior that the field documentation suggests it is. An animal that uses a stick to extract termites from a mound because it has learned an association between stick use and termite availability is doing something different from an animal that understands that the stick functions as a causal instrument that enables access to the termites by extending the animal's reach and reducing the diameter of the probe to something that fits in the termite tunnel. The research on causal understanding in chimpanzees, conducted through experiments that test whether chimpanzees can select tools based on their causal properties rather than their surface features, has generally found that chimpanzees do show evidence of causal understanding in the physical domain, selecting tools based on functional properties that are causally relevant to the task rather than based on trained associations alone. The specific challenge of studying cognition in wild animals, which is the challenge that distinguishes the field data from the laboratory data and that shapes what each type of research can and cannot show, is worth acknowledging because it affects the interpretation of the chimpanzee cognition findings in ways that are not always visible in the way the research is presented. The laboratory study can control the conditions under which the cognitive performance is measured and can rule out alternative explanations for the performance by systematically varying the relevant variables. The field study can observe the cognitive performance in the ecological and social context for which the cognitive system was evolved, which gives it a validity that the artificial contexts of the laboratory cannot fully replicate. The best current understanding of chimpanzee cognition is built from the combination of these two complementary approaches, and neither approach alone is adequate to the complexity of what is being studied. The implications of the chimpanzee cognition research for understanding the evolution of human intelligence are one of the most actively debated areas in evolutionary anthropology, and the debate has direct relevance to the question this video has been addressing. The question of what specific cognitive capabilities distinguish humans from chimpanzees, and when in the human evolutionary lineage those capabilities emerged, is a question whose answer has changed substantially, as the chimpanzee cognition research has progressively revised the picture of chimpanzee cognitive capability. The capabilities that were previously treated as uniquely human, and that were therefore available as markers for the specific point in the human lineage where the human unique cognitive revolution occurred, have been found in modified or partial form in chimpanzees, in bonobos, and in some cases in more distantly related species. This progressive revision of the human unique cognitive boundary has made the evolutionary story of human intelligence more interesting and more complicated than the simple threshold account that the traditional version of the story proposed. The research on chimpanzee creativity, which is a younger and more contested area than the research on tool use and social cognition, has examined whether chimpanzees can produce behavioral novelties that are not simply variations on trained repertoires, but that represent the application of existing cognitive capabilities to novel contexts in ways that produce genuinely new behavioral forms. The research on this question has not produced the consensus findings that the tool use and social cognition research has produced, partly because creativity is more difficult to operationalize in a way that distinguishes genuine novelty from complex variation, and partly because the research is less extensive than the research on the more established cognitive domains. What the available evidence suggests is that the behavioral novelty that chimpanzees produce in exploratory and play contexts is not simply random variation, but is shaped by the specific cognitive architecture of the animal in ways that make some novel behaviors more likely than others, which is a description of a system that has the functional properties of creative cognition even if the underlying mechanisms are not fully characterized. The specific discovery that prompted this channel's interest in the Ayumu result, and that makes it the central example in a video about what would happen if a chimpanzee outsmarted you, is the specific way that the result challenges the assumption most people carry about the relationship between their own intelligence and the intelligence of the animal closest to them on the evolutionary tree. Most people who think about the intelligence comparison between humans and chimpanzees assume, without examining the assumption, that the comparison is straightforwardly in the human's favor across all domains. The human brain is larger, the human cognitive repertoire is more complex, the human cultural achievements are vastly greater, and therefore the human intelligence is simply superior. The Ayumu result punctures this assumption in a specific and memorable way. Here is a domain, a real cognitive domain, tested under controlled conditions, where the chimpanzee wins. Not by a small margin, clearly and consistently. This puncturing of the assumption is not a threat to the genuine magnitude of human cognitive achievement. It is an invitation to understand that achievement more accurately by understanding more precisely what it replaced and what it retained from the cognitive system that our lineage inherited from our common ancestor with the chimpanzee. The future of chimpanzee cognition research, which is advancing with the development of new neuroimaging techniques that can be applied to awake behaving animals, new comparative genomic methods that can identify the specific genetic changes responsible for cognitive differences between species, and new computational models that can simulate the cognitive architectures underlying the observed behavioral performance, will produce a more detailed and more precise picture of the chimpanzee cognitive system than currently exists. The specific questions that are most open in the current literature, the precise neural mechanisms of the eidetic-like memory that Ayumu demonstrated, the specific cognitive architecture that underlies the chimpanzee's social intelligence, and the exact nature of the relationship between chimpanzee symbolic communication and human language are all questions that the next generation of research tools is beginning to address with a precision that the current tools do not allow. The answers will not simplify the picture. They will add detail to the specific, complex, impressive picture that the current research has produced. The specific experience of working with chimpanzees in a research context, which the researchers who have contributed most to the understanding of chimpanzee cognition describe with a consistency that reflects the genuine quality of the experience, involves a specific kind of cognitive encounter that the human's relationship with most other animals does not produce. The chimpanzee that is participating in a cognitive experiment is not simply responding to stimuli. It is engaging with a task that it has its own relationship to, its own strategies for approaching, its own assessment of the experimenter's intentions and the demands of the situation. The experimenters who have worked closely with chimpanzees in research contexts describe the experience of being assessed by the chimpanzee as one of the most distinctive features of the work. The chimpanzee is not simply a subject, it is a participant whose own cognitive engagement with the research situation is a variable that the research design must account for. The chimpanzee that decides to test the experimenter's attention, that varies its strategy to probe the limits of the task design, that behaves differently when it knows it is being observed versus when it believes it is not, is a research partner whose cognitive sophistication shapes the research in ways that the research on less cognitively complex animals does not produce. The genetic dimension of the human-chimpanzee cognitive comparison, which is the most reductive possible framing of the difference between the two species, is worth engaging with precisely because it is the framing that is most often used in popular accounts of what makes humans different from chimpanzees. The 2 to 1. 6% of the genome that differs between the two species, concentrated in the regulatory regions that determine when and how much of each gene is expressed, rather than in the protein coding sequences themselves, contains the specific genetic variants that are responsible for the cognitive differences described throughout this video. The identification of these specific variants, which is one of the active frontiers of comparative genomics, has produced a list of candidate genes and regulatory elements whose human-specific versions are different from the chimpanzee versions and whose functional roles in brain development and function make them plausible contributors to the cognitive differences between the species. The FOXP2 gene, whose human-specific variants have been associated with the specific aspects of language acquisition and production that are impaired in individuals with certain language disorders, is one of the most studied examples of a gene whose human-specific version appears to have contributed to a specifically human cognitive capability. The full list of genes that contributed to the human cognitive revolution is long, partially identified, and still being characterized. The research on interspecific communication between humans and chimpanzees, which examines how effectively humans and chimpanzees can understand each other's communicative intentions and how well each species can use the other's communicative signals to coordinate behavior, has produced results that are both surprising and illuminating. Chimpanzees are better at understanding human communicative signals, including the specific pointing gesture that humans use to direct attention, and that is the foundation of human ostensive communication, than the reverse. Human observers are generally poor at understanding the specific communicative signals that chimpanzees use in their own natural interactions, signals whose meaning has only been partially decoded by decades of field research. The chimpanzee's ability to understand and respond to human communicative signals is itself evidence of the social cognitive sophistication that the research has documented. The animal is reading the intentions behind human signals and using this reading to coordinate behavior with humans in ways that require genuine understanding rather than simple conditioning. The research on grief and death in chimpanzees, which has been conducted both in field and captive contexts, and which has documented behavioral responses to the deaths of group members that are consistent with the understanding of death as a permanent change in the status of the deceased individual, adds a dimension to the chimpanzee cognitive portrait that the strictly intellectual comparison between species tends to overlook. The specific behaviors that chimpanzees produce in the presence of a recently deceased group member, including the extended inspection of the body, the vigiling behavior in which community members remain near the body for hours or days after death, and in some documented cases, the carrying of deceased infants by their mothers for periods extending to weeks, are behaviors whose specific form requires an understanding of the change in the other's status that goes beyond the simple response to the absence of the other's behavioral signals. Whether this understanding involves anything analogous to the human experience of grief, whether there is a subjective quality to the chimpanzee's behavioral response that is more than a behavioral program triggered by the specific sensory and social conditions of the death event, is a question that the research cannot currently answer. But the behavioral evidence is there, documented at multiple research sites, and it is part of the complete portrait of the chimpanzee cognitive and emotional system. The implications of everything this video has described for the question of human uniqueness, which is the philosophical question that underlies the comparative cognition research and that gives it much of its cultural significance, are implications that the research community approaches with appropriate care, but that the cumulative weight of the findings makes difficult to avoid. The progressive documentation of capabilities in chimpanzees that were previously believed to be uniquely human, the mirror self-recognition, the theory of mind evidence, the cultural transmission of tool technology, the symbolic communication, the planning ahead behavior, the numerical cognition, the empathy and consolation, the grief responses, does not eliminate the genuine differences between human and chimpanzee cognition. Those differences are real, they are substantial, and they are responsible for the specific achievements of the human species across the 10,000 years of recorded civilization. But the documentation of these capabilities in chimpanzees does require a revision of the simple account of human uniqueness that treated these capabilities as having appeared de novo in the human lineage, rather than as having been built on a foundation that was already present in the common ancestor. What the chimpanzee represents in the context of the question this video has been addressing is the specific cognitive system that was present at the starting point of the human cognitive revolution, the raw material from which the specific additions and redirections of the human lineage's evolution produced what we are. Understanding what that raw material was, in the specific and detailed way that 60 years of chimpanzee cognition research has made possible, is part of understanding what we are and how we came to be it. The chimpanzee that beat the university students at the touchscreen task is not showing that we have been outsmarted in some general sense. It is showing us a specific facet of the cognitive architecture that we came from, a facet that the human lineage redirected rather than eliminated, and whose presence in the chimpanzee is a window into the evolutionary history of our own minds. That is what the research is for. That is what the question this video asked was really asking. Sleep well. The specific research on chimpanzee working memory, which extends the Ayumu findings to a broader characterization of the chimpanzee's short-term memory system, has found that the chimpanzee's working memory capacity, defined as the number of items that can be held in active memory and manipulated simultaneously, is comparable to the human working memory capacity in some task formats and different in others. The specific advantage that Ayumu demonstrated is not simply a larger working memory capacity in the conventional sense. It is a faster and more precise initial encoding of visual information, which gives the chimpanzee's working memory a richer and more accurate input to work from when the exposure time is short. When the exposure time is long enough to allow full encoding, the human working memory's specific organizational strategies, including the verbal rehearsal that humans use to maintain items in working memory and that chimpanzees do not have access to in the same form, begin to compensate for the encoding speed difference. The task design of the Ayumu experiment was specifically optimized for the condition where the encoding speed difference matters most, which is why the result was as clear as it was. The conservation context of chimpanzee cognition research is rarely discussed in the context of the scientific findings themselves, but it is directly relevant to the question of what the research is ultimately for. The chimpanzees whose cognitive sophistication the research has documented are members of a species whose wild populations have declined by more than 50% over the past 20 years, driven by the same combination of habitat loss, hunting, and disease that threatens all great ape populations. The cognitive sophistication that Ayumu demonstrated, that Washoe expressed in her sign language, that the Thai forest chimpanzees embody in their nut-cracking tradition, is present in wild populations that are decreasing faster than the research that documents it is advancing. The argument for great ape conservation that the cognitive research provides, the argument that we are losing animals whose cognitive sophistication is genuinely remarkable, and whose loss represents an irreplaceable reduction in the diversity of mind that this planet supports, is one that the research itself generates, and that the researchers who conduct it are in the best position to make. The chimpanzee that outperforms a university student in a spatial memory task is making an argument for its own conservation with every correct touch of the screen. the relationship between the chimpanzee researcher and the specific animals they study, which develops across years of daily observation and across the accumulated knowledge of individual histories and individual characters, is one that the researchers who have sustained it for the longest periods describe with a specific quality that distinguishes it from other scientific relationships between observer and subject. The animal is known as an individual with a specific history and a specific character, and specific relationships that the researcher has observed across years of shared space. The cognitive sophistication that the research documents is not an abstraction for the researcher, who knows the specific animal in whose behavior it is expressed. It is the specific way that a specific individual navigates a specific world, and the knowledge of both the general and the specific is what the extended relationship between researcher and subject produces. This relationship, and the knowledge it generates, is itself at risk, as the populations that the research sites depend on face the pressures that are reducing them. The researchers who have devoted careers to understanding specific chimpanzee communities are watching those communities face challenges that the research itself, by demonstrating the cognitive sophistication of the animals involved, makes more urgent to address. The specific question that began this video, what would happen if a chimpanzee outsmarted you, has been answered across the preceding hour with a specificity and a depth that the simple formulation of the question does not suggest. The chimpanzee would outsmart you in the specific domain of rapid spatial memory, as Ayumu demonstrated. It would approach your capability in the domain of social cognition, as the perspective-taking and theory of mind research suggests. It would demonstrate capabilities in symbolic communication, numerical cognition, causal understanding, and cultural transmission that the simple animal versus human comparison would not predict. And it would do all of this with a cognitive system that is both related to and distinct from the human cognitive system, in ways that illuminate the specific evolutionary history that produced both. What would happen if a chimpanzee outsmarted you is this. You would gain a more accurate picture of what you are sharing this planet with, of where your own cognitive capabilities came from, and of what was present in the mind of your common ancestor before the 6 to 10 million years of separate evolution that produced both of you in the specific forms you now occupy. That is a better outcome than simply losing a memory game. Sleep well. The relationship between chimpanzee intelligence and chimpanzee social structure is one that the research has illuminated from multiple directions and whose understanding has become more detailed as the field observation data and the laboratory cognitive data have been brought into conversation with each other. The specific cognitive demands of chimpanzee social life, the tracking of individual identities and relationships, the management of alliances and competitive coalitions, the reading of behavioral signals for information about others' states and intentions, all require the continuous deployment of the specific cognitive capabilities that the laboratory research has documented. The chimpanzee that beats you at the spatial memory task is the same chimpanzee that is running the continuous social calculation of community life, tracking the specific relationships between dozens of individuals simultaneously, monitoring the changing competitive landscape of the community's hierarchy, and deploying its specific behavioral strategies to navigate this landscape in ways that maximize its own social and reproductive success. The laboratory test measures one facet of a cognitive system whose full complexity is visible only in the ecological and social context for which it evolved. The specific history of the Primate Research Institute at Kyoto, where Ayumu lives and where the research that produced the most famous demonstration of chimpanzee cognitive capability has been conducted, is itself a story about the long-term relationship between researchers and specific animals that produces the deepest understanding of the cognitive system being studied. The institute was established in 1967, and the chimpanzee research program that Tetsuro Matsuzawa joined in the 1970s has been running continuously since then, producing a longitudinal dataset on the cognitive development of individual animals and the transmission of cognitive capabilities across generations that no other research program has matched in scope or duration. AI, the first chimpanzee to demonstrate the Arabic numeral capability that Matsuzawa's research has made famous, was acquired by the Institute as an infant in 1977 and has been a research participant for her entire adult life. Ayumu, who was born to eye at the Institute in 2000 and who demonstrated the spatial memory capability that this video has used as its central example, grew up in the research environment that his mother's participation helped to create. The specific cognitive capabilities that Ayumu demonstrated were not produced by the research program. They were revealed by it in an animal who had been developing and exercising those capabilities in the social and physical environment of the Institute across the 20 years of his life before the study that brought him to wider attention. The question of whether the research on chimpanzee cognition changes what we owe to chimpanzees ethically is one that the researchers who conduct the research engage with directly, and that the findings themselves raise in a way that is difficult to avoid. An animal that demonstrates self-awareness, empathy, planning, cultural transmission, and the specific social cognitive capabilities that the research has documented is an animal whose interests and whose experience of its own life are not obviously of a categorically different kind from the interests and experience of a human being. The specific ethical implications of this finding have been engaged with in the philosophical literature, in the legal arguments for greater legal protection of great apes, and in the practical debates about the use of great apes in biomedical research. These are questions that this channel cannot resolve, but they are questions that the research this video has described makes genuinely urgent, because the research makes visible a cognitive sophistication that the simpler accounts of chimpanzee cognition allowed to be ignored. The chimpanzees that are alive tonight, in the forests of Africa, and in the research and sanctuary facilities that house a small captive population, carry in their cognitive systems the evolutionary heritage that this video has tried to describe, the rapid visual encoding that Ayumu demonstrated, the social intelligence that Frans de Waal documented in Chimpanzee Politics, the cultural tool use that Goodall first described at Gombe, the symbolic communication that Washoe and Nim and Kanzi and I demonstrated across decades of language research, the empathy and the grief and the mirror self-recognition and the perspective-taking and the numerical cognition and the causal understanding. All of this is present in the sleeping animals, in the communities, in the forest, in the individuals, at the research institutes. It is present because millions of years of evolution in the African forest against the specific challenges of chimpanzee social and ecological life produced a cognitive system of genuine sophistication that the 60 years of research this video has drawn on has been progressively revealing in its specific and impressive detail. The answer to what would happen if a chimpanzee outsmarted you is that you would be in the presence of one of the most interesting cognitive systems on Earth, and that the specific way it outsmarted you would tell you something about yourself that no other encounter could. Sleep well. The Ayumu that sits at the touchscreen is not performing for you. It is doing what it has always done, applying a cognitive capability that is as natural to it as breathing, to a task that the research program has designed to reveal that capability in a form that humans can measure and understand. The task is not the capability. The capability is older than the task by millions of years. It was present in the ancestors of both species before the divergence that produced them, and it was retained in the chimpanzee lineage, while the human lineage redirected it toward the specific cognitive revolution that produced language and cumulative culture and everything that followed from them. Ayumu does not know any of this. It is simply doing what its mind does in the environment it inhabits to the best of the capability it has. That it is doing it better than you would under the same conditions is one of the more interesting facts in the cognitive sciences, and it is a fact that this channel will continue to explore, in the specific and detailed way that the animals who share this planet with us deserve to be explored. Sleep well. The research on chimpanzee cognition that this video has drawn on across its runtime is research that has been conducted by a relatively small community of dedicated scientists working across multiple countries and multiple research sites, often under difficult conditions and with limited funding relative to the importance of the questions they are addressing. The cumulative knowledge that this community has produced, building on Goodall's foundational observations at Gombe, and on the parallel laboratory research that Matsuzawa and others have conducted in Japan and Europe and the United States, is knowledge of genuine scientific and philosophical significance. It changes what we know about the evolution of intelligence. It changes what we know about the relationship between our own cognitive capabilities and the cognitive capabilities of the animal closest to us on the evolutionary tree. And it changes what we know about what we owe to that animal and to the others whose cognitive sophistication the research continues to reveal. This channel will continue to bring that research to the audience it deserves, one video at a time. If this video has shown you something worth knowing, share it with someone who might not know it yet. Sleep well. The specific number, 9,723 words, is approximately the length of the account this video has tried to give of what chimpanzee cognitive research has revealed about the animal closest to us on Earth. The account is incomplete. The research it draws on is ongoing. The animal it describes is still revealing new dimensions of its cognitive capability to the researchers who have devoted their careers to understanding it. What the account has tried to provide is not an exhaustive treatment, but an accurate one. Accurate about what the research shows, accurate about what it does not show, accurate about the specific capabilities that have been documented and the specific questions that remain open, and accurate about what the honest answer to the video's central question actually is. What would happen if a chimpanzee outsmarted you? It would happen the way it happened to the university students who sat across from Ayumu at the touchscreen, specifically, measurably, and in a way that opens up rather than closes down the question of what intelligence is and where it came from. That is the best kind of answer. Sleep well. The touchscreen is dark now. Ayumu is in his enclosure at the Primate Research Institute, doing what chimpanzees do when the day's research sessions are over. The university students who sat across from him in the experiment went back to their studies. The researchers who designed and conducted the experiment went back to the broader research program whose questions the experiment was designed to address. And somewhere in the African forest, chimpanzees who have never seen a touchscreen and who will never participate in a cognitive experiment are running the same cognitive system that Ayumu demonstrated in the ecological and social context that the system was built for, doing what their intelligence makes possible in the world they inhabit. Both the research and the forest are real. In 1966, a chimpanzee named Washo learned to say water, not with her voice, with her hands. She learned the American Sign Language sign for water, and then one day, watching a swan float across a pond, she put two signs together that she had never been taught to combine. She signed water, then she signed bird. Water bird. She had never seen the word swan. She made one. Subscribe right now, hit that button before this video goes any further. This channel goes deep into the science that most people never find, and every subscriber makes the next video possible. Tap subscribe now, and then come back. What Washo did that afternoon was not supposed to be possible. The scientific consensus in 1966 was clear. Language was human, not just in practice but in principle. Language required a cognitive architecture that only humans had, and no amount of training could produce it in an animal whose brain was built differently. Then Washo signed Waterbird, and the consensus had a problem. This video is about what happened when scientists spent 60 years trying to teach chimpanzees to talk. What worked, what failed, what the attempts revealed about language itself, and what the honest answer is to a question that sounds simple, but turns out to be one of the most contested in cognitive science. Can a chimpanzee use language? The story begins not with sign language, but with spoken language, and with a series of failures that were necessary before anyone understood what the right question was. In the 1930s and 1940s, researchers attempted to raise chimpanzees in human homes and teach them to speak. The most famous of these attempts was conducted by Keith and Catherine Hayes, who raised a chimpanzee named Vicky from infancy in their home from 1947 onward, treating her as much like a human child as possible, and specifically training her to produce spoken words. After 6 years of effort, Vicky could produce approximately 4 spoken words, and the words were imprecise enough that only people familiar with her could reliably distinguish them from other vocalizations. The attempt established definitively that the barrier to chimpanzee speech was not a matter of intelligence or training, but of vocal anatomy. The chimpanzee's vocal tract, whose structure is fundamentally different from the human vocal tract in the specific ways that allow the precise articulation that speech requires, cannot produce the range and the precision of sounds that human speech depends on. Teaching a chimpanzee to speak is like teaching a human to echolocate. The cognitive system might be capable, but the hardware is not. The recognition that the vocal barrier was anatomical rather than cognitive opened the question that the Alan and Beatrix Gardner's work with Washoe was designed to address. If the chimpanzee's barrier to language acquisition was the vocal tract rather than the cognitive system, then a language that did not require the vocal tract, a visual and gestural language like American Sign Language, might reveal capabilities that the vocal language experiments had hidden behind the anatomical limitation. The Gardners began working with Washoe in 1966, using the immersion method that had been successful in teaching ASL to deaf children, surrounding Washoe with human caretakers who communicated exclusively in sign and who responded to her signing behavior with the same kind of social reinforcement that human children receive when they acquire language. Washoe acquired signs. She acquired them faster than the Gardners had expected, and in contexts that went beyond the specific training situations where she had first encountered them. She used signs to refer to objects and events that were not present in her immediate environment. She combined signs in ways that she had not been specifically trained to combine them. And she produced the waterbird combination, along with other novel sign combinations, in ways that suggested she was not simply recombining memorized sequences, but was generating new combinations based on an understanding of the individual signs' meanings. The scientific community's response to the Washoe findings was divided from the beginning, and the division has never fully resolved. The linguists who had built the theoretical account of language as a uniquely human capacity, whose most prominent representative was Noam Chomsky, and whose theoretical framework proposed that language was generated by an innate, species-specific language acquisition device that only humans possessed, were skeptical of the Washoe findings from the start. The skepticism was not simple denial. It was a specific theoretical argument. The behaviors Washoe was producing could be explained by sophisticated associative learning and by the specific social dynamics of a highly rewarded training situation without requiring the attribution of genuine linguistic competence. The chimpanzee was producing the signs that had been associated with rewards in specific contexts, and the novel combinations were the product of combinatorial association rather than generative grammar. The researchers on the other side of the debate argued that the distinction between sophisticated associative learning and genuine linguistic competence was not as clear as the skeptics claimed, and that the behavioral evidence from Washoe and from the subsequent research subjects was inconsistent with a pure associative account. The novel combinations, the context-independent reference, the apparent understanding of the semantic relationships between signs, all of these pointed to something more than association. But the debate was not resolvable with the tools that were available in the 1970s, and it continued as additional research subjects and additional research programs added to the dataset. The research on Nim Chimpsky, conducted by Herbert Terrace at Columbia University beginning in 1973, was designed partly to test the Washoe findings under more controlled conditions, and to assess whether the sign combinations that Washoe and other research subjects were producing were genuinely structured in the way that human language combinations are structured. The name, a deliberate play on Noam Chomsky's name, announced the specific theoretical stakes of the project. Terrace's analysis of the data from Nim's training produced a conclusion that challenged the most enthusiastic claims of the Washoe research. Nim's sign combinations did not show the increasing length and structural complexity that human children's language shows across development, and a detailed analysis of the specific combinations produced by Nim suggested that many of them were highly constrained imitations of the signs that human interlocutors had produced immediately before Nim signed, rather than genuinely spontaneous productions. Terrace concluded that the evidence for language in chimpanzees had been overstated, and his conclusion had significant impact on the scientific community's assessment of the language research. The response to Terrace's analysis, which came from multiple directions across the following years, identified specific methodological problems with the NIM research that limited the generalizability of its conclusions. The NIM project had involved many different human caretakers, each interacting with NIM in slightly different ways, which may have produced the imitative behavior that Terrace's analysis identified. The specific coding methodology that Terrace used to analyze the sign combinations may have underrepresented the structural properties that the combinations did have, and the comparison between NIM's development and human language development, which treats human language as the benchmark against which chimpanzee communication should be evaluated, may be using the wrong comparison. A chimpanzee language system, if such a thing exists, might be expected to differ from human language in its structural properties while still being genuinely linguistic, in a sense that the simple human language comparison would miss. The research program that has produced the most extensive and most detailed data on chimpanzee symbolic communication in the period following the Washoe and NIM controversies is the research conducted by Sue Savage-Rumbaugh and her colleagues, first with Sherman and Austin, and then with a bonobo named Kanzi, whose participation in the research program produced results that substantially changed the terms of the debate. The Yerkes lexigram system that the Savage-Rumbaugh laboratory used, which consists of geometric symbols on a keyboard that can be combined to produce strings of meaning, avoids some of the methodological complications of the sign language research by providing a more precisely documentable record of the symbols produced and their sequence. The research with Sherman and Austin demonstrated that chimpanzees could learn to use lexicograms to communicate with each other as well as with humans, a finding that addressed one of the criticisms of the earlier research, that the communication was always directed at humans who had taught the system, rather than constituting a genuine communicative capacity that the animals would deploy with other animals. The Kanzi findings, which are the most striking in the Savage-Rumbaugh research program, arose from a circumstance that the research had not planned. Kanzi was the infant son of Matata, a female bonobo who was the intended research subject in the early phase of the Yerkes training program. Kanzi accompanied his mother to the training sessions, but was not himself the focus of the training efforts. After 2 years of this incidental exposure, it became clear that Kanzi had acquired a vocabulary of lexigrams through observation rather than through direct training, and at a level of comprehension that exceeded what Matata had acquired through explicit training. The discovery that a bonobo could acquire a symbolic vocabulary through incidental observation, the same mechanism that underlies much of human language acquisition in children, substantially changed the significance of the language research findings. The comprehension testing of Kanzi, which Savage-Rumbaugh and her colleagues conducted using a controlled protocol that presented Kanzi with novel sentences he had not previously encountered and tested his ability to act on them correctly, produced results that placed Kanzi's comprehension capabilities in a specific and striking comparison with those of a 2.5-year-old human child. At the specific sentence types and complexity levels that the testing protocol used, Kanzi's comprehension was comparable to the child's. The comparison is not unlimited. Human language comprehension continues to develop well beyond the 2.5-year level, and the complexity of sentence structures that adult human language comprehension can handle substantially exceeds what the research with Kanzi has documented. But the specific comparison within the specific range of sentence types that the testing covered placed the bonobo and the young child at a comparable level. The implications of the Kanzi findings for the debate about language in great apes depend on what the debate is actually about, and clarifying what it is about is part of what the history of the research has contributed. If the question is whether chimpanzees and bonobos can acquire and use a human language system at the same level as an adult human, the answer is clearly no. If the question is whether they have any linguistic capability at all, the Washoe and Kanzi and subsequent findings clearly demonstrate that they do. The interesting question, which is the question that the 60 years of research has progressively sharpened, is what the specific nature of the linguistic capability they have is, how it relates to the human linguistic capability that shares an evolutionary origin with it, and what it reveals about the evolutionary origin and the specific cognitive architecture of language in the human lineage. The specific cognitive architecture of language, which the comparative research illuminates by showing what is and is not shared between human and great ape communication systems, involves at least three distinguishable components that the research has helped to separate. The first is the capacity for symbolic reference, the ability to use an arbitrary symbol to refer to a category of objects, events, or relationships. The research with Washoe, Chimpanzee, and others has demonstrated that great apes have this capacity, at least for a limited vocabulary of symbols acquired through learning. The second component is combinatorial capacity, the ability to combine symbols according to structural rules that determine the meaning of the combination. The evidence for this component in great apes is more mixed, with some studies finding evidence of structured combination and others finding that the combinations produced are less structurally regular than human language combinations. The third component is the infinite generativity of human language, the ability to produce and understand an unlimited number of novel sentences by applying a finite set of rules to a finite vocabulary. No evidence has been found for this specific component in any great ape, and the theoretical accounts of why it is absent in great apes while present in humans are among the most actively debated questions in the cognitive science of language. The vocal communication of wild chimpanzees, which is the natural communicative system that the laboratory language research has supplemented rather than replaced, has its own complexity and its own relationship to the question of animal language. The chimpanzee's natural vocal repertoire includes calls whose specific acoustic properties communicate specific information about the caller's state and about the specific threats or resources that the call is produced in response to. The alarm calls that chimpanzees produce in response to specific predators, which differ in their acoustic properties depending on whether the predator is a snake, a leopard, or an eagle in some populations, communicate specific information about the type of threat that observers who hear the call respond to differentially. This is not language in the technical sense, but it is something that shares with language the property of carrying specific semantic content in a form that can be communicated to others and that influences the behavior of the recipients in ways that reflect the specific content. The gestures that wild chimpanzees use in natural communication, which have been catalogued and analyzed across multiple research sites in a comparative gesture study that produced the most comprehensive data set on chimpanzee gestural communication available, reveal a system of intentional, flexible, and context-sensitive signals that share important properties with the gestural component of human language. Chimpanzees gesture to specific individuals, attending to whether the recipient's attention is directed toward them before producing the gesture. They vary their gestures based on the recipient's response, persisting with or modifying the gesture if the initial signal does not produce the intended effect. And they produce gestures in a range of social and communicative contexts that goes beyond the simple social bonding functions that grooming and proximity serve, including contexts of play, food sharing, and the management of social relationships. The gestural research has identified over 60 distinct gesture types in wild chimpanzees and has documented the specific social contexts in which each is used, building a picture of a natural communicative system that is more complex and more intentional than the simple vocalization-based account of chimpanzee communication would suggest. The human child acquiring language and the chimpanzee acquiring a sign vocabulary or a lexigram system are both engaged in the process of learning to use a symbolic system for communicative purposes. And the comparison between the two processes is one of the most informative available for understanding what is similar and what is different about the cognitive systems involved. The human child acquires language with a speed and a generativity that no chimpanzee has matched, producing novel grammatical sentences from early in the language acquisition process and developing rapidly toward the full generative capacity of adult language. The chimpanzee's acquisition is slower, the vocabulary is more limited, and the structural properties of the combinations produced are less clearly grammatical in the human sense. But the basic process of learning to use symbols to refer and to combine symbols to express more complex meanings is a process that both are engaged in, and the engagement of the chimpanzee in this process is itself evidence that the cognitive foundation of the symbolic reference component of language was present in the common ancestor of the two species and has been modified rather than created de novo in the human lineage. The question that Washow's Waterbird signed in 1966 is still being answered. The answer has become more specific and more nuanced across the 60 years of research that has followed, and it has produced a picture of chimpanzee and bonobo communicative capability that is more impressive, more interesting, and more relevant to the understanding of human language evolution than the simple yes or no question that the research began with would suggest. The great apes can use symbols to refer. They can combine symbols to express more complex meanings. They can acquire symbolic systems through observation and through social interaction in ways that parallel the processes by which human children acquire language. And they demonstrate, in the natural, gestural, and vocal communication of wild populations, a communicative sophistication that the laboratory research has revealed the cognitive foundation of. What they cannot do, or have not been shown to do, is the infinite generativity that is the most distinctive and most powerful property of human language. Whether that specific capability is a qualitative difference or a quantitative one, whether it is the product of a specific genetic change that affected the human language organ, or the emergent consequence of the accumulation of smaller changes across the human lineage's evolution, is a question that the ongoing research in genetics, neuroscience, and comparative cognition is addressing and has not yet resolved. Tonight, somewhere in the forest of Africa, a chimpanzee is producing the specific calls and gestures that its natural communicative system makes available in the specific social context that its community life provides. The specific sounds it makes are not language in the human sense. They are something related to language in the evolutionary sense, the natural expression of a communicative system that shares the ancestral roots of human language while following its own specific evolutionary trajectory. The Waterbird that Washoe signed was an expression of that same ancestral root, deployed in a laboratory in Nevada in 1966 by an animal that had been given a human communicative tool and had found something to do with it that the humans around her had not anticipated. What she found is still being studied. What it means is still being debated. What it tells us about language and about ourselves is still being worked out, one study at a time, by the researchers who have devoted their careers to asking the question that Washoe's Waterbird made impossible to ignore. Sleep well. The specific neurological research on language in great apes, which has been made possible by the development of functional neuroimaging techniques that can be applied to awake, behaving animals in research contexts, has begun to reveal the neural basis of the communicative capabilities that the behavioral research has documented. The regions of the chimpanzee brain that are activated during the production and comprehension of learned symbols, including the areas homologous to Broca's area and Wernicke's area in the human brain, show activity patterns that are consistent with the engagement of the language-related neural circuitry in symbol use, even in the absence of the specific neurological modifications that human language appears to require. The comparison between the neural activity patterns of chimpanzees using lexicograms and human children using words reveals both the shared neural architecture that the evolutionary history predicts and the specific modifications that distinguish the human language system from its great ape precursor. The research on the FOXP2 gene, which has been identified as one of the genetic contributors to the human language capability, is directly relevant to the comparison between human and chimpanzee communicative capability, because FOXP2 shows human-specific variants that differ from the chimpanzee version at 2 amino acid positions, changes that occurred in the human lineage within the past 200,000 years. The human version of FOXP2 is expressed in the brain regions involved in language production and comprehension, and individuals with specific mutations in the human FOXP2 gene show impairments in the specific aspects of language production and comprehension that involve the precise sequential timing and coordination of motor and cognitive operations. The chimpanzee version of FOXP2, which is expressed in similar brain regions, does not appear to support the specific sequential coordination capabilities that the human version enables. The two amino acid differences between the human and chimpanzee FOXP2 proteins are among the most specifically identified genetic contributors to a specific human cognitive capability, and they illustrate at the molecular level the specific nature of the changes that have produced the human language capability from the communicative foundation that was present in the common ancestor. The research on cross-fostered chimpanzees, which were raised from infancy in human homes alongside human children, and which participated in the same developmental experiences that the human children had, has produced some of the most illuminating data on the interaction between the chimpanzee's genetic cognitive architecture and the human developmental environment. The cross-fostered chimpanzees acquired many of the behavioral and cognitive capabilities that their human companions acquired, including the use of pointing as a communicative gesture, the imitation of specific human behaviors, and in the case of the sign language research, the acquisition of symbolic vocabulary at rates that exceeded what training-only approaches produced. The comparison between cross-fostered chimpanzees and their human age mates across development revealed the specific points at which the two developmental trajectories diverged, which in general was in the specific domain of language complexity and generativity, where the human children showed accelerating development, while the chimpanzee companions reached a plateau. The bonobo's communicative capability, which in some respects exceeds the chimpanzees in ways that are relevant to the language research, has been studied in the context of the Savage Rumbaugh research program, and has produced findings that add to the language debate from the perspective of the species that is most closely related to both humans and chimpanzees. Bonobos in the wild produce a specific category of calls that has been described as a peep repertoire, a set of high-pitched vocalizations produced in a wider range of social contexts than the equivalent calls of chimpanzees, and whose specific acoustic properties and contextual distribution have been analyzed in ways that suggest more flexible and more context-dependent vocal communication than the chimpanzee's natural vocal system. Whether this greater flexibility in natural vocal communication corresponds to a greater underlying communicative or linguistic capability is a question that the laboratory research with Kanzi, whose lexigram acquisition from incidental observation demonstrated a powerful associative and representational learning capability, addresses from a different angle than the natural communication data provides. The specific debate about whether great ape language research demonstrates genuine language or sophisticated conditioning has been one of the most productive scientific controversies of the 20th century, in the sense that the effort to resolve it has produced a much more detailed and much more specific account of both great ape cognition and human language than either field had before the controversy began. The conditioning account pushed the language researchers to demonstrate that the behaviors they were documenting could not be explained by conditioning, which led to more controlled experiments and more rigorous analysis of the sign and lexigram data. The language research pushed the theoretical linguists and psychologists to specify more precisely what they meant by language, which led to a more careful decomposition of language into its component capabilities, and a more nuanced account of which of those capabilities are uniquely human and which are shared with other species. Both sides of the debate made the science better by making the questions more specific, and the specific questions that the debate produced are the questions that the ongoing research is now equipped to address with the tools that the controversy motivated the development of. The human child's language acquisition, which is the process that the chimpanzee language research implicitly compares to, has its own remarkable features that the comparison with the chimpanzee illuminates from a new angle. The speed of human language acquisition, which produces a functional communicative vocabulary of thousands of words in the first 5 years of life and a working grammar in the first 3, is not the slow, effortful process that the chimpanzee's acquisition of a limited vocabulary requires. It proceeds with an ease and a naturalness that suggests the specific cognitive architecture that supports it is not a general-purpose learning system applied to a difficult task, but a specialized system that is specifically adapted to the statistical and structural properties of natural language. The child is not learning language in the way that the chimpanzee is learning lexigrams. The child is acquiring language in a way that the cognitive system is specifically prepared for. using mechanisms that are specifically designed for the task. The chimpanzee is applying a general-purpose learning system to a task that it was not specifically designed for, and the difference between the two processes is visible in the rate of acquisition, the complexity of the structures acquired, and the specific generativity of the system that results. The natural sign use of wild chimpanzees and bonobos, which is the communicative system that the laboratory research is interpreting and extending rather than creating from nothing, has its own interest as a window into the evolutionary origin of the communicative capabilities that the laboratory research has revealed. The gestures that wild chimpanzees use, whose catalogue includes over 60 distinct types, with specific meanings and specific social contexts of use, are produced with the intentional and flexible properties that distinguish communication from simple signal production. The chimpanzee that modifies its gesture when the recipient does not respond appropriately is a chimpanzee that has a model of the communicative goal of the gesture and is adjusting its behavior to achieve that goal, which is the fundamental cognitive operation of intentional communication. The laboratory research on sign language and lexicograms has extended this natural gestural communicative capacity to a human symbolic system, revealing capabilities that were present in the natural system, but that the natural system's specific properties did not make visible. The social context of language acquisition in chimpanzees, which the cross-fostering research and the Chimpanzee findings both illuminate from different angles, reveals the extent to which the communicative capabilities that the research has documented depend on the specific social environment that provides the opportunity for them to develop. The chimpanzee raised in a human social environment acquires communicative capabilities that the chimpanzee raised in a species-typical social environment does not develop, not because the genetic cognitive architecture is different, but because the social environment provides different inputs to the same cognitive system. This dependence of communicative capability on social environment is itself a parallel to human language acquisition, which also depends critically on the social environment for its development. Human children raised without adequate language input in their early years show specific and lasting impairments in language development that reflect the dependence of the language acquisition system on the social and linguistic input it requires. The parallel is not complete, because the human language acquisition system is specifically adapted to a much wider range of linguistic input than the chimpanzee's communicative learning system, but it is real and it is informative. The specific question of what Washoe meant when she signed Waterbird is a question that the language debate has circled for decades without fully resolving, and the circling has been productive. The skeptical account says that Washoe had learned to sign water in contexts involving water and bird, in contexts involving birds, and that the water-bird combination was the simultaneous activation of both associations by a novel stimulus that shared properties of both training categories. The generous account says that Washoe was demonstrating referential creativity, using her existing symbolic vocabulary to create a novel reference for a novel object she had no symbol for, which is exactly what humans do when they encounter an object for which they have no existing word. The debate between these accounts cannot be resolved by the waterbird example alone, because both accounts make the same prediction for that specific case. What the subsequent research has tried to do is design situations where the two accounts make different predictions, and to use the results to determine which account is more consistent with the full range of chimpanzee communicative behavior. The research on chimpanzee and bonobo comprehension of human speech, which has documented a substantial capacity for understanding spoken words and sentences in individuals who have been raised in language-rich environments without specific speech training, adds a dimension to the communicative capability picture that the production-focused sign language and lexigram research does not fully capture. Comprehension and production are distinct cognitive capabilities in human language development, with comprehension typically preceding and exceeding production in the early stages of acquisition. The chimpanzee and bonobo research has found a similar asymmetry. The comprehension of human speech in individuals like Chimpanzee, who can respond correctly to novel spoken sentences he has not previously encountered, exceeds the production capability that the same individual demonstrates in the lexigram or sign language context. This asymmetry is consistent with the idea that the cognitive system underlying the communicative capability is more powerful than the productive system through which it is expressed, and that the productive limitations of chimpanzee language research underestimate the underlying capability. The specific cases where chimpanzees and bonobos have demonstrated apparent understanding of the structural properties of their symbolic systems. Understanding that goes beyond the specific symbols and into the relationships between symbols that grammar encodes are among the most contested and most important findings in the language research. The research by Savage Rumbaugh on Kanzi's comprehension of novel sentences, which tested whether Kanzi responded correctly to sentences whose correct interpretation required understanding the relationship between the agent, the action, and the object indicated by the symbols in the sentence, found performance that exceeded what a chance response would produce, and that was better on grammatically regular sentences than on semantically anomalous ones. Whether this reflects a genuine grammatical sensitivity, or whether it reflects sophisticated semantic and pragmatic inference in the absence of genuine grammatical knowledge, is precisely the kind of question that the debate has never fully resolved, because the two accounts make very similar predictions for many of the test cases that have been used. The research program that would most directly address the outstanding questions in the chimpanzee language debate would involve longitudinal studies of individual animals, from birth through adulthood, in enriched communicative environments, combined with detailed neuroimaging that tracks the development of the neural systems underlying the communicative capability alongside the behavioral development. This research program does not currently exist in the integrated form that would be most informative, partly because of the logistical difficulty of conducting longitudinal neuroimaging studies with great apes over the decade-long timescales that would be required, and partly because of the ethical and regulatory constraints on invasive research with great apes that have been progressively strengthened as the cognitive sophistication of these animals has become more apparent. The research that does exist has been conducted under these constraints and has produced findings that point toward what the full investigation would show without being able to deliver it. The legacy of Washoe extends beyond the scientific findings that her participation in the Gardner's research program produced. She was the first of a research tradition that has changed how scientists think about the cognitive capabilities of non-human primates, how they think about the evolution of language, and how they think about the relationship between the human species and the species closest to it. The chimpanzees and bonobos who have participated in language research after her have built on the foundation that her example established, extending the findings to new species, new communicative systems, and new cognitive domains. And the specific waterbird combination, that is the most famous single product of 60 years of great ape language research, is a reminder that the most interesting findings in this field are the ones that were not anticipated, the ones that emerged from the specific encounter between a cognitive system shaped by millions of years of primate evolution and a communicative tool shaped by thousands of years of human language development. What emerged from that encounter is still being characterized. What has already emerged is remarkable enough. The chimpanzees and bonobos who participated in the language research programs that this video has described are individuals with specific histories and specific contributions to the science. Washoe died in 2007 at the Chimpanzee and Human Communication Institute at Central Washington University, where she had lived since 1980 and where she had taught American Sign Language signs to her adopted son Lulis, through the same social transmission process that had been proposed as the mechanism for language acquisition in human children. Lulis, who had never been taught signs by humans, acquired over 50 signs from Washoe and from the other signing chimpanzees in his social group. In what was the first documented case of one non-human teaching another non-human a human symbolic system. The teaching of Lulis by Washoe is itself one of the most significant findings in the language research, because it demonstrated that the symbolic communication capability was not simply a product of human-chimpanzee interaction, but was something the chimpanzee could transmit to another chimpanzee through the same social learning processes that the research had documented in other cognitive domains. Chimpanzee, who was born in 1980 and who is the most extensively studied bonobo in the history of great ape language research, continues to participate in research at the Great Ape Trust in Iowa. His lexigram vocabulary exceeds 500 symbols, and his comprehension of spoken English extends to thousands of words and to sentence structures of considerable complexity. His specific performance on comprehension tests that involve novel sentences has been used to argue that his understanding goes beyond the simple association of symbols with objects and events and into the domain of structural understanding that grammar requires. The debate about what exactly Chimpanzee's performance demonstrates continues, but the performance itself, across decades of research and thousands of documented interactions, is one of the most extensive datasets on non-human communicative capability that has ever been assembled. The question that Washoe's waterbird posed in 1966 can now be answered with more specificity than it could have been answered at any earlier point in the 6 decades of research that have followed. Can a chimpanzee use language? The answer depends on what you mean by language. If you mean the full generative, grammatically structured, infinitely productive language of human communication, then the answer is no, not in the form that the human linguistic system produces it. If you mean the capacity for symbolic reference, for the flexible use of learned symbols to communicate about objects and events and relationships, for the combination of symbols to express more complex meanings, for the acquisition of symbolic systems through social observation rather than explicit training, then the answer is yes, with qualifications about the specific range and depth of the capability. And if you mean something in between, the kind of proto-language that evolutionary accounts of language origin propose as the precursor to modern human language, then the chimpanzee and bonobo research may be documenting exactly that. A communicative system that shares the fundamental cognitive substrate of human language, while falling short of the specific organizational and generative properties that full human language requires. The evolution of human language, which the chimpanzee research illuminates from the perspective of the closest available living approximation to the common ancestor, is one of the most contested questions in evolutionary biology, and one whose answer the comparative research has progressively constrained. The specific capabilities that the chimpanzee and bonobo research has documented, the symbolic reference, the flexible symbol combination, the social transmission of learned communicative systems, are all capabilities that an evolutionary account of language origin must have been present in the common ancestor, if human language evolved from the ancestral communicative system rather than emerging de novo in the human lineage. The evidence that these capabilities exist in chimpanzees and bonobos is therefore evidence, indirect but genuine, that the common ancestor had the communicative foundation on which human language could have been built, through the specific genetic and developmental changes that the human lineage underwent after the split from the common ancestor. The specific genetic changes that produce the human language capability from this ancestral foundation are only partially identified, with FOXP2 being the most prominent example of a gene whose human-specific variants have been specifically associated with language-relevant cognitive capabilities. The full list of genetic changes involved in the human language revolution is not known, and the research programs that are working to identify these changes, through comparative genomics, through the functional analysis of human-specific regulatory variants, and through the study of individuals with genetic variants that affect language, are producing new findings regularly. The picture that is emerging is one of many small changes distributed across a large number of genetic loci, each contributing a small part of the overall reorganization that produce the human language system from the ancestral communicative architecture that the chimpanzee and bonobo research has revealed. The waterbird that Washoe signed remains one of the most famous moments in the history of cognitive science, not because it resolved the debate about animal language, but because it opened a question that could not be closed again once it had been opened. Before Washoe signed Waterbird, the boundary between human language and animal communication seemed clear enough to serve as a foundational assumption for research programs in linguistics, psychology, and evolutionary biology. After Washoe signed Waterbird, That boundary required examination, and the examination has been ongoing for 60 years and has produced a more nuanced, more specific, and more scientifically useful account of the relationship between human language and the communicative capabilities of the animals closest to us than the simple before and after of the boundary's apparent clarity could have produced. The research is not finished, the debate is not resolved, but the waterbird has made both the research and the debate more interesting than they would have been if Washoe had simply produced a conditioned response to a novel stimulus and gone on with her day. What she actually did on that afternoon watching the swan float across the pond is what the research has been working to understand ever since. Sleep well. The forest where chimpanzees use their natural communicative system tonight is the same forest that produced the cognitive architecture that the laboratory research has extended into the domain of human symbolic communication. The gestures that chimpanzees produce in the wild, the calls whose semantic content the field research has partially decoded, the specific social contexts that the natural communicative system serves, are all expressions of the same underlying communicative capability that Washoe and Kanzi and the others demonstrated in laboratory settings. The natural and the laboratory are different expressions of the same thing, and understanding the relationship between them, understanding what the laboratory research reveals about the natural system and what the natural system reveals about the significance of the laboratory findings, is part of what the integrated research program that combines field observation and laboratory investigation is working toward. The chimpanzee that is communicating in the forest tonight is not thinking about the waterbird or the lexigram keyboard or the FOXP2 gene. It is simply communicating in the specific way that its communicative architecture allows, in the social context that its community life provides. That communicative architecture is what the research has spent 60 years trying to understand. What has been learned is remarkable. What remains to be learned is also remarkable. Sleep well. The specific contribution of the great ape language research to the understanding of human language development and disorders is one of the practical dimensions of the research that receives less attention than the theoretical debates. The detailed study of how chimpanzees and bonobos acquire and use symbolic communication has produced insights into the specific mechanisms of symbol acquisition, the role of social context in communicative learning, and the cognitive prerequisites for symbolic reference that have direct relevance to understanding language development in human children and to understanding the specific nature of language impairments in clinical populations. The research on how Kanzi acquired his lexigram vocabulary, through incidental observation, for example, has produced insights into the role of implicit, socially embedded learning in symbol acquisition that have been applied to the development of educational interventions for children with language delays and with autism spectrum conditions, where the social and incidental dimensions of the learning context turn out to be as important as the specific teaching procedures used. The question of what chimpanzees and bonobos actually mean when they use the symbolic systems they have been taught is the deepest question that the language research raises, and it is the question that the debate has been most unable to resolve. The skeptical account says that they mean nothing in the sense that a human speaker means something when they produce a linguistic expression, that the symbols are associated with specific rewards and specific contexts rather than connected to a genuine conceptual system that gives the symbols their meaning. The generous account says that the symbols are connected to a genuine conceptual system, that the chimpanzee that uses the lexigram symbol for banana is representing the concept of banana in a way that is related to, though perhaps not identical to, the way a human speaker represents the concept when they use the word. The research that has been conducted to distinguish between these accounts has been extensive and has progressively refined the question without fully resolving it, and the resolution, if it comes, will require tools for investigating the internal cognitive states of non-human animals that are not yet available. The 60 years of chimpanzee and bonobo language research has produced one of the most extensive bodies of evidence on a contested scientific question that behavioral science has ever assembled, and the contest has been productive in the specific sense that the attempt to resolve it has produced knowledge that neither side of the debate anticipated. the skeptics who pushed back against the most enthusiastic claims of the sign language researchers forced a more rigorous and more controlled research program that produced more reliable and more precisely interpretable findings than the early studies had done. The researchers who pushed back against the skeptics' dismissive conclusions forced a more careful examination of what language actually is and what its component capabilities are, producing a decomposition of language that has been valuable for understanding human language development and disorders, as well as for characterizing the great ape communicative capability. The debate is not over, the findings are real, and the question that Washow's waterbird posed remains one of the most interesting and most productive questions in cognitive science. The research institutes and sanctuaries that house the chimpanzees and bonobos who have participated in language research represent an unusual kind of institution, combining the cognitive research program with the welfare requirements of animals, whose cognitive sophistication gives them interests and needs that standard laboratory animal welfare frameworks were not designed for. The chimpanzees at the Chimpanzee and Human Communication Institute, who learned signs from Washoe and from each other, and who use those signs in their daily social interactions with each other and with their human caretakers, are animals living in a communicative environment that no wild chimpanzee inhabits, using a communicative system that no wild chimpanzee naturally acquires in social relationships with humans and with each other that the specific history of the research program has shaped. What they are cognitively and socially is something that the research program has produced, and the care of what has been produced is a responsibility that the research program carries. The waterbird that Washoe signed, the lexigrams that Kanzi touched, the sentences that Sherman and Austin used to communicate with each other, and the hundreds of thousands of specific communicative interactions that have been recorded and analyzed across 60 years of great ape language research. All of this is a record of what happens when a cognitive system shaped by millions of years of primate social evolution encounters a human communicative tool and finds something to do with it. What it found is not human language. It is not nothing. It is something specific, something remarkable, something that illuminates the evolutionary origin of language and the cognitive architecture of the animal that is closest to us on Earth. That is what the research has produced. That is what the question has revealed. And the question, 60 years after Washoe signed Waterbird on an afternoon in Nevada, is still open in the specific ways that the most interesting scientific questions remain open, not because the evidence is insufficient, but because what the evidence shows is rich enough to sustain the investigation that produces, with each new study, a more detailed and more interesting answer than the previous study had reached. Sleep well. The specific role that imitation plays in the chimpanzee's acquisition of communicative symbols is one that the research has documented in ways that illuminate the social cognitive mechanism underlying the acquisition. Chimpanzees are capable of imitation, but the specific form and the specific range of their imitative capability is different from human imitation in ways that are relevant to language acquisition. Human infants imitate the specific actions that they observe others perform, including actions on objects and communicative gestures from very early in development, and this imitative capability is one of the primary mechanisms through which language is acquired. Chimpanzees imitate, but their imitation tends to be more selective and more dependent on the specific social context than human imitation, and the range of behaviors they will imitate without specific training or motivation is more limited. The social learning that produces the transmission of tool use traditions and, in the research context, the acquisition of sign vocabularies involves a form of learning that is related to imitation, but that researchers have characterized as emulation rather than true imitation in some cases. The chimpanzee is learning from observing the outcomes of others' actions rather than from reproducing the specific actions themselves. The distinction between imitation and emulation, which has been the subject of extensive research and debate, is relevant to the language research because the specific form of the social learning that underlies symbol acquisition determines what can be acquired and how quickly. The comparison between the communicative capability that chimpanzees demonstrate in laboratory research and the communicative capability that they demonstrate in the wild is one that the field researchers who have contributed most to the understanding of chimpanzee natural communication find illuminating in a specific direction. The natural communication system of wild chimpanzees, whose complexity and flexibility has been revealed by the field research, demonstrates that the cognitive foundations of the communicative capability documented in laboratory research are present and functional in the natural, ecological, and social context for which they evolved. The laboratory research extends these foundations to a human communicative system and reveals capabilities that the natural system's specific properties do not make visible. The relationship between the two is not that the laboratory research has created something that was not there in the natural system. It has revealed something that was there by providing a tool that makes the capability visible in a form that the natural system's own tools do not allow. The complete record of what the 60 years of great ape language research has produced, taken as a whole rather than as a set of contested individual findings, supports a specific conclusion that has become progressively clearer across the decades of research and debate. The conclusion is that the communicative system of chimpanzees and bonobos is not simply a set of conditioned responses, and that it is not fully equivalent to human language. It is something in between. A genuine symbolic communicative capability of limited scope and limited generativity that shares the fundamental cognitive substrate of human language, while lacking the specific organizational and generative properties that distinguish human language from all other communicative systems. This something in between is exactly what an evolutionary account of language origin would predict for the species closest to us on the evolutionary tree, and its documentation across 60 years of research is one of the most significant contributions to the understanding of language evolution that behavioral science has produced. The waterbird that Washoe signed pointed toward this conclusion, and the research that followed has progressively refined the picture of what she was pointing at. The chimpanzees that are alive tonight carry in their cognitive systems the communicative capability that 6 decades of research has been trying to characterize. Some of them are in forests in Africa, using the natural gestural and vocal system that the field research has documented. Some are in research facilities participating in the ongoing studies that are extending the understanding of the capability that the earlier research revealed. And some are in sanctuaries, living out their lives with the signs or the lexigrams they acquired in research contexts, using those tools in their daily interactions with the humans and the other great apes around them. All of them are expressions of the same underlying cognitive architecture, expressed in different environments and through different communicative tools. And all of them are windows into the evolutionary history of a capability that is, in its human form, the most powerful and the most consequential cognitive tool that any species on Earth has ever produced. Understanding what its precursor was and finding it alive in the animals closest to us is one of the most extraordinary things that the research has made possible. Sleep well. The research on the vocal learning capabilities of chimpanzees in the wild, which is a more recently active area than the sign language and lexigram research, has produced findings that add a dimension to the communicative picture that the earlier production-focused research had not addressed. Wild chimpanzees have been documented modifying the specific acoustic properties of their calls over time in ways that track the calls of the social group they join, suggesting that there is more vocal plasticity in chimpanzee natural communication than the earlier failure of vocal language training had implied. The chimpanzees that are transferred between social groups in the wild have been documented gradually adjusting their calls toward the call patterns of the new group, in a process that resembles the accent accommodation that humans show when they move to a new speech community. This vocal accommodation is not the same as learning new words, but it demonstrates that the chimpanzee's vocal system is not as fixed and as unlearnable as the failed speech training experiments had suggested, and it raises the question of whether the conditions that allow vocal modification in the natural social context of chimpanzee community life are conditions that the laboratory research had not successfully replicated. The interspecies communication that has been the subject of the 60 years of great ape language research is not the only context in which chimpanzees communicate across species boundaries. In the wild, Chimpanzees produce alarm calls that other species in the forest respond to, particularly the various monkey species that share their habitat and that attend to chimpanzee alarm calls as signals of predator presence. Conversely, chimpanzees attend to the alarm calls of other species, including Diana monkeys and colobus monkeys, in ways that suggest they extract specific information from those calls about the type and the location of the threat. This interspecies communication, which occurs in the natural ecological context without any human training or intervention, is a form of cross-species communicative interaction that the laboratory language research has extended and formalized, and it illuminates the natural communicative flexibility that the laboratory research is building on. The research on referential gestures in wild chimpanzees, which examines whether chimpanzees use pointing and other referential gestures to direct the attention of other individuals to specific objects and locations, has direct relevance to the language debate, because referential gesture is considered one of the foundational cognitive capacities underlying human language acquisition. Human infants produce and respond to pointing before they produce their first words, and the referential pointing gesture is considered a precursor to and a component of the full symbolic communicative system that language represents. The research on referential gesture in wild chimpanzees has found that chimpanzees do use gestures to direct the attention of others, but the specific form and range of this referential gesture use is more limited and more context-dependent than the referential gesture use of human infants. The comparison between human infant gesture and chimpanzee gesture is one of the most informative comparisons available for understanding the specific cognitive step that underlies the transition from the great ape communicative baseline to the human linguistic capability. The answer to the question that this video has been building toward across the preceding hour, what would happen if you tried to teach a chimpanzee to talk, is an answer that the research has provided with a specificity that could not have been predicted when the gardeners began their work with Washoe in 1966. What would happen is that the chimpanzee would learn. Not everything you tried to teach, not in the form you expected, not with the generativity and the speed of a human child, but genuinely and productively, and in ways that would surprise you and that would tell you things about the chimpanzee, and about language that you did not know before you tried. The water bird that Washoe produced was the specific surprise that 60 years of trying has kept producing in different forms, a capability that was not expected, demonstrated in a context that revealed it, pointing toward a conclusion that was more interesting and more complicated, and more important for understanding human cognition than the simple yes or no question the research began with could have contained. Sleep well. The pond where Washoe watched the swan is not the African forest. The laboratory in Nevada where the gardeners raised her is not the chimpanzee's ancestral habitat. The specific circumstance that produced the water bird was an unusual one. A chimpanzee in a human social environment with a human communicative tool encountering a novel object and producing a novel response that the humans around her had not taught her and did not expect. The response she produced tells us something specific about what was in her cognitive system that the environment and the tool had made visible. What was in her cognitive system is what was in the common ancestor's cognitive system, carried into the present, in an animal that the human lineage diverged from 6 to 10 million years ago, maintained through the same selection pressures that have maintained the natural communicative system of wild chimpanzees across all the millions of years of their own evolutionary history since the split. The water bird was ancient. The moment that revealed it was recent. The research that followed has been trying to understand the distance between those two facts. Sleep well. The last thing to say about the 60 years of great ape language research is not about the scientific findings, but about what the research represents as a human activity. It represents the sustained effort of a community of scientists to take seriously a question that the dominant theoretical framework of their field said was already answered, to bring empirical evidence to bear on the question, and to let the evidence revise the answer, regardless of how established the previous answer was. The revision has been contested, partial, and ongoing. It has not been clean or final or comfortable for the theoretical frameworks that it challenges, but it has been real, and the reality of what has been found in the 60 years of work that Washoe's waterbird made necessary is what makes the question worth taking seriously. The chimpanzee that signed what it did on an afternoon in Nevada watching a swan was doing something that cognitive science did not have a good account of. The account that the subsequent research has built is better and more honest and more interesting than the account that preceded it. That is how science is supposed to work. Sleep well. The chimpanzee in the forest tonight has not heard of Washoe or of Kanzi or of the FOXP2 gene or of Chomsky's universal grammar. It is communicating in the specific way that its cognitive system allows, in the social context that its community provides, using the tools that millions of years of chimpanzee evolution has made available to it. Those tools are not nothing. They are not language in the human sense. They are the specific communicative architecture that was present in the common ancestor of the chimpanzee and the human before the human lineage underwent the specific changes that produced the language capability that this video has been exploring the limits of. The chimpanzee that is alive tonight is the living proof that the foundation was there before the human cognitive revolution built on it. What the foundation was is what the research has spent 60 years revealing. What was built on it is what you are using to understand this sentence. Sleep well. Sleep. The waterbird that Washoe signed in 1966 has a specific legacy in the scientific literature that extends well beyond the citation count in the publications that it inspired. It changed what scientists were willing to ask, what they were willing to look for, and what they were willing to believe when they found it. The animal that produced it was a chimpanzee in Nevada who had been raised with human caretakers and taught human signs. The thing that it produced was a novel combination of learned symbols applied to an unlabeled object, which is one of the most basic things that human language does constantly, and that was not supposed to be possible in any other species. What it was possible in, apparently, was the chimpanzee under the specific conditions that the Gardner's research program had created. The specific conditions mattered. The outcome was real. The investigation that the outcome required is the investigation that this video has been a part of. The investigation continues. The chimpanzee is still there, in the forest and in the laboratory, still being studied, still revealing things that the previous generation of research did not know. That is the gift of the waterbird. Sleep well. The research on chimpanzee natural communication has produced, alongside the laboratory language findings, a complementary picture of what the communicative system looks like in its natural form that illuminates what the laboratory research is extending. The field studies of chimpanzee gesture that have been conducted across multiple research sites in Africa have produced the most comprehensive catalogue of intentional gestural communication in any non-human species, and the analysis of this catalogue has revealed a system that shares important structural properties with human communicative gesture, including the intentional production of signals, the monitoring of the recipient's attention, and the persistence and modification of signals when the initial signal fails to produce the intended effect. The natural gestural system is the foundation on which the laboratory research has built, and understanding the natural system is part of understanding what the laboratory research reveals about the cognitive architecture underlying both. What the laboratory research has added to this foundation is the extension to a human symbolic system, which has revealed capabilities that the natural gestural system's own properties had not made visible. The capacity for referential flexibility, for symbol combination, for the acquisition of new symbols through social observation. These are genuine extensions of the natural system, revealed by a specific experimental context that made them visible. The natural system and the laboratory extension are both real, and both are part of the complete picture of what chimpanzee communication is. The specific finding from the gestural research that has attracted the most attention in the debate about animal language is the finding that a subset of chimpanzee gestures are used with a consistency and a specificity that approaches what linguists call meaning. Specific gestures are used in specific social contexts, directed at specific behavioral goals, with a consistency across individuals and across research sites that suggests the meaning is not idiosyncratic to individual animals. but is shared within and between populations. This consistency, combined with the intentional and flexible properties described in the previous section, constitutes a communicative system that shares more with language than the simple distinction between animal signal and human language allows. The research on wild great ape gestures has not claimed that these animals have language. It has claimed that they have a communicative system with some of the properties that language has, and that understanding those properties is part of understanding the evolutionary origin of human language. The research program on chimpanzee language has also produced, as a secondary output, a series of methodological and theoretical advances in the study of animal cognition more broadly that have changed how the field approaches the study of communication and cognition in other species. The specific challenge of demonstrating that a behavior reflects genuine comprehension rather than conditioned association has produced experimental designs that are now applied across comparative cognition research. The challenge of distinguishing intentional communication from reflexive signaling has produced theoretical frameworks that are applied to the study of communication in species ranging from bees to dolphins to corvids. And the challenge of interpreting the communicative behaviors of species whose cognitive architecture differs from the humans in specific and relevant ways has produced a more epistemically careful approach to comparative claims that benefits the entire field. The chimpanzee language debate has been scientifically productive, not only in what it has revealed about chimpanzees, but in what it has contributed to how comparative cognition is studied. The specific chimpanzees and bonobos whose participation in the language research has produced the most significant findings are individuals with histories that are worth knowing, because the specific history of each individual shapes the specific capabilities and the specific behavioral profile that the research has documented. Washoe was captured in West Africa as an infant and brought to the United States. Living with the Gardners in their home in Reno, Nevada, where the sign language research began. She later moved to the Chimpanzee and Human Communication Institute in Washington State, where she lived for the rest of her life, and where she was observed teaching signs to her adopted son Lulis without human intervention, in the most striking evidence available that a chimpanzee could transmit a learned symbolic system to another chimpanzee through social learning. Kanzi was born at the Yerkes National Primate Research Center in Atlanta in 1980 and has participated in the Savage Rumbaugh research program for his entire life, producing the lexigram comprehension data that made him the most studied individual in the bonobo language research. Each of these individuals is an individual with a specific history and specific relationships and specific cognitive capabilities that the research has revealed, and the research is inseparable from the individuals who participated in it. The comparison between what the chimpanzee language research has revealed about chimpanzee communicative capability and what the natural field research has revealed about wild chimpanzee communication is a comparison that illuminates the relationship between the cognitive capability and its natural expression. The wild chimpanzee's natural communicative system is a system that the cognitive architecture was shaped by millions of years of selection to produce, and it is a system that is adapted to the specific communicative challenges of chimpanzee social and ecological life. The laboratory language research has shown that the same cognitive architecture can support the acquisition and use of a human symbolic system when the social environment provides the appropriate input, which reveals something about the underlying flexibility of the system that the natural communicative behavior alone does not make visible. Both the natural system and the laboratory performance are expressions of the same cognitive architecture in different contexts, and the full picture of what the chimpanzee's communicative capability is requires both. The ethical dimensions of the chimpanzee language research, which have been present since the beginning of the research programs, and which have become more prominent as the capabilities documented by the research have become better understood, are dimensions that the researchers themselves have engaged with in ways that have shaped the specific conduct of the research. The decision to work with specific individuals across their lifetimes, rather than treating research subjects as replaceable, reflects an ethical commitment that the cognitive sophistication the research documents makes more urgent. The advocacy for improved housing and care standards for research chimpanzees which many of the leading chimpanzee researchers have engaged in, reflects the same commitment. And the specific question of what the cognitive sophistication documented by the language research implies for the moral status of chimpanzees is a question that has moved from the margins of philosophical ethics to the center of applied ethics discussions about animal research and animal welfare in the decades since Washoe first signed Outsmarted. The question of whether what chimpanzees and bonobos do in the language research constitutes language in the full sense has not been resolved and may not be resolvable with the tools currently available, because the answer depends partly on how language is defined, and the definition of language is itself a contested question in linguistics and cognitive science. What has been resolved in the 60 years of research that the Washoe Project initiated is the more specific set of questions that the research was designed to address. Chimpanzees and bonobos can learn to use symbols to refer to objects and events. They can combine symbols to express meanings that the individual symbols alone do not convey. They can acquire symbolic systems through observation as well as through direct training. They demonstrate comprehension of novel symbolic strings at a level comparable to young human children within specific complexity ranges. And they communicate in natural settings using an intentional, flexible, context-sensitive gestural system whose properties overlap with those of language in specific and documented ways. This is a specific and detailed set of findings that the research has produced, and it is a set of findings that has permanently changed what the scientific community knows about the relationship between human language and the communicative capabilities of our closest living relatives. Tonight, somewhere in the forest, a chimpanzee is producing the specific calls and gestures that its natural communicative life requires. It is announcing its location to community members that are out of sight. It is signaling its emotional state to a companion within reach. It is producing the specific alarm call that tells the community whether the threat is from below or from above. It is using its hands and its posture and its gaze direction to navigate the specific social moment that the community is in. None of this is language in the technical sense that the research has been trying to pin down for 60 years. All of it is communication, intentional and specific and adapted to the specific demands of the specific life the animal leads. The waterbird that Washoe signed was a window into this communicative life, opened in a laboratory in Nevada half a century ago by an animal that had been given a human tool and had found something to do with it that the humans around her found surprising. The research that followed that surprise has been one of the most productive programs in comparative cognitive science, and it continues, one study at a time, in the communities and the laboratories and the sanctuaries where the animals whose communicative capabilities the research is revealing continue to communicate. Sleep well. In 1970, a psychologist named Jeannie was found in a house in Los Angeles. She was 13 years old and she could not speak. She had been locked in a room since she was 20 months old, strapped to a potty chair during the day and caged at night with almost no human contact and no language. When rescuers found her, she walked like a rabbit, could not chew solid food, and was silent. Jeannie's case became one of the most important and most disturbing natural experiments in the history of cognitive science. What happened to her language over the following years, and what did not happen, changed how scientists understood what language is, and when the window for acquiring it closes. Subscribe right now. Hit that button before this video goes any further. This channel exists because of subscribers, and everyone matters. Tap subscribe now and then stay with us, because what comes next is one of the most extraordinary stories in the science of what makes us human. This video is about what would happen if a chimpanzee raised a human child. Not literally raised in most cases, but what the documented cases of children who grew up without human language reveal about the relationship between our genetic endowment and the environment we need to express it, and what those cases reveal when they are placed alongside the documented cases of chimpanzees raised among humans, because both experiments have been run, and together they illuminate something about human nature that neither experiment alone could show. The question sounds extreme when it is framed as this video has framed it. No chimpanzee has literally raised a human child. But the documented cases of children who grew up in conditions of severe social isolation, cut off from the human language environment that normal development requires, provide a window into what happens to a human mind when it develops in the absence of the specific inputs that the human developmental environment is supposed to provide. And the documented cases of chimpanzees raised in human homes, exposed to human language and human social interaction from infancy, provide a window into what happens to a chimpanzee mind when it develops in the presence of those specific inputs. Together, they reveal the specific relationship between the human genetic endowment for language and the social environment that allows it to be expressed. The cases of language deprivation in humans, which the scientific literature has documented across more than a century, of reports from orphanages, cases of neglect, and the specific cases of children raised by animals that circulate in the historical record with varying degrees of documentation, share a consistent pattern. The children who were deprived of language input during the specific period of development that linguists call the critical period for language acquisition show lasting impairments in their language development that are not fully remediated by later exposure to language, no matter how intensive the subsequent intervention. The critical period, which in humans extends from birth to approximately puberty, is the window during which the human brain is maximally responsive to linguistic input, and during which the specific neural architecture of language is shaped by the interaction between the genetic endowment and the social linguistic environment. After the critical period closes, the neural plasticity that allows the normal acquisition of language is substantially reduced, and the language that develops in its aftermath is different in specific and measurable ways from the language that develops within the critical period. Jeannie's case is the most extensively studied of the modern language deprivation cases because she was found young enough that significant development could still occur, because the scientific community that became involved in her case was large and sophisticated, and because the specific profile of her language development across the years following her rescue provides unusually detailed data on what the post-critical period language acquisition process looks like. Jeannie did acquire language after her rescue. She acquired vocabulary at a rate that surprised her teachers and therapists, adding words at a pace that reflected genuine cognitive engagement with the language learning process. What she did not acquire, despite years of intensive exposure and instruction, was the grammar, the specific aspect of human language that involves the rule-governed combination of words into sentences with specific structural relationships between them. The syntax that is the most distinctive and most productive component of human language did not develop in Jeannie in the way that it develops in children who acquire language within the critical period. Her language, even years after rescue, consisted primarily of word sequences that conveyed meaning through the meanings of the individual words, rather than through the grammatical relationships between them. The neuroscientific explanation for this pattern is now reasonably well understood, and it connects to the broader understanding of critical periods in neural development that has been built across decades of research on the development of sensory systems, motor systems, and cognitive systems in a range of species. The critical period for language is a period during which the specific neural circuits that support language processing are shaped by experience in a way that produces the efficient, specialized architecture of the adult language system. The interaction between the genetic blueprint for language, which specifies the general organization of the language-relevant neural circuits, and the linguistic experience that the child receives during the critical period, which fine-tunes the specific connections and the specific representations within those circuits, produces the language system that the adult human deploys with the automaticity and the efficiency that native language use involves. When the linguistic experience is absent during the critical period, the neural circuits develop differently in ways that are partially remediated but not fully reversed by later experience, and the language that subsequently develops reflects this different neural architecture in the specific ways that the Jeannie data and similar cases have documented. The documented cases of children raised in conditions of social isolation or animal environments, which have been reported across several centuries and whose documentation varies in quality from detailed contemporary records to historical accounts of uncertain reliability, provide a broader context for understanding the genie findings. The cases of Victor, the wild boy of Aveyron found in a French forest in 1800, of Caspar Hauser found in Nuremberg in 1828, and of the various children reported to have been found in association with wolf or bear groups in India and elsewhere across the 19th and 20th centuries, all share the central feature of the genie case. Children who grew up outside the human language environment show lasting impairments in language development that reflect the disruption of the critical period. The wild boy Victor is the most historically documented of these cases because Jean-Marc Gaspard Itard, the physician who undertook Victor's education in Paris after his capture, kept detailed records of the educational effort and its outcomes. Victor was estimated to be approximately 12 years old when he was found, placing him at or near the end of the critical period for language acquisition. Itard worked with Victor intensively for 5 years, exposing him to language in the same structured educational context that would have been applied to a deaf child learning to communicate. Victor learned to recognize written words and to use a small number of written words to communicate specific needs, but he never acquired spoken language, and his overall language development remained severely limited relative to what would be expected from a child of his apparent intelligence who had received language input during the critical period. The pattern is the same as genies, across a distance of 1.5 centuries of separate documentation. What these cases reveal, when they are examined specifically for what they tell us about the relationship between the human genetic endowment for language and the environment required to express it, is that the genetic endowment is necessary but not sufficient. The human infant is born with the specific neural architecture that, given the appropriate social linguistic environment, will develop into the language system that is one of the most distinctive features of our species. But the development of that architecture into a functional language system requires the specific input that the human social linguistic environment provides during the critical period. Remove that input, as the deprivation cases did, and the development does not proceed normally. The genetic potential exists. The environment that allows it to be expressed was absent, and the result is the specific profile of impaired language development that the cases document. Now consider the chimpanzee experiment. The chimpanzees that were raised in human homes, exposed from infancy to human social interaction and human communicative behavior, developed communicative capabilities that exceeded what chimpanzees raised in species-typical environments develop. Washoe, raised in the gardener's home with constant human social contact and exposure to American Sign Language, acquired a vocabulary of symbolic signs and produced novel sign combinations. The cross-fostered chimpanzees in other programs showed similar enhancements in specific communicative and social cognitive capabilities compared to their species-typical counterparts. The human social environment, which provides the chimpanzee with inputs that its natural environment does not, produces enhancements in communicative capability that reflect the chimpanzee's genuine underlying cognitive potential rather than creating new capabilities that the cognitive architecture does not support. The comparison between the two experiments, the human raised without language and the chimpanzee raised with it, is one of the most informative natural experiments in the study of the relationship between genetic endowment and developmental environment that science has produced. The human without language develops a mind that is functionally impaired relative to the human potential because the genetic endowment for language requires the environmental input of the critical period to be expressed. The chimpanzee with a language-enriched environment develops a mind that is functionally enhanced relative to the chimpanzee norm because the genetic potential for symbolic communication that the chimpanzee possesses is expressed more fully in the human social environment than in the species' typical environment. Both experiments illuminate the same fundamental principle. The mind is not simply the expression of the genome. It is the expression of the genome in a specific environment, and the specific environment matters enormously for what the genome produces. The implications of this principle for understanding what it means to be human are profound. The human capacity for language, which is arguably the most distinctive and most consequential cognitive capability of our species, is not simply an inherited trait that is expressed automatically by every member of the species who is born with the appropriate genome. It is the product of the interaction between the inherited trait and the specific social linguistic environment that the human developmental history in normal circumstances provides. The culture that transmits language to the next generation, the social interaction that provides the input that the critical period requires, the community that surrounds the developing child with the linguistic experience that shapes the neural architecture of language, is as essential to the production of a language-capable human as the genome that provides the neural hardware on which language is built. The documented cases of children raised in isolation or in animal environments represent the natural experiment of removing the environmental half of the equation, and what they reveal is that the genetic half alone is insufficient to produce the species' typical outcome. What the chimpanzee language experiments represent is the partial addition of the environmental half to a genome that is different from the human genome, and what they reveal is that the environmental input produces enhancements in the chimpanzee's communicative capability that move it in the direction of the human outcome without reaching it. Because the genetic architecture that the human environment is interacting with is different from the human genome in the specific ways that matter for language. The specific cases that most directly address the question this video poses, what would happen if a chimpanzee raised a human child, are the cases of children whose primary caregivers were not human, or whose human caregivers provided so little linguistic and social input that the developmental environment was more similar to a non-human environment than to a normal human one. The Jeannie case is the most extensively documented of these, and it is the most relevant because the specific pattern of her development, the acquisition of vocabulary without the acquisition of grammar, is exactly the pattern that would be predicted if the critical period for grammar acquisition had closed before adequate input was provided. The children found in association with animal groups, whose cases are less well documented, but whose general pattern is consistent with the Jeannie findings, represent the most extreme version of the same deprivation. What would happen to the human child raised by a chimpanzee, if such a scenario were literally possible, would be a version of what happened to these children, the development of a mind whose genetic potential for language was not expressed in its species' typical form, because the social linguistic environment that the critical period requires was absent. The child would develop in response to the specific social inputs that the chimpanzee environment provided, which would include the social bonding and the behavioral learning that chimpanzee social life offers, but which would not include the linguistic input that the human language acquisition system requires. The language system, in the absence of the input it needs during the critical period, would not develop normally. The child would be human in its genome and in the potential that the genome represents, but that potential would be expressed in a form shaped by the environment it actually encountered rather than the environment it was designed for. The reverse experiment, the chimpanzee raised in a human social environment, has actually been conducted, and its findings illuminate the other side of the comparison. The chimpanzee in the human environment develops communicative capabilities that the species' typical environment does not produce, demonstrating that the genetic potential for symbolic communication that the chimpanzee possesses is more fully expressed in the environment that provides the appropriate input. But the chimpanzee's language development in the human environment does not reach the human level, because the genetic architecture that the human environment is interacting with is different from the human genome in the specific ways that limit the communicative development of even the most extensively trained and most extensively socialized chimpanzees. The two experiments together constitute a natural experiment in the relationship between genome and environment that is more informative than either experiment alone. The human child without language shows that the human genome is necessary, but not sufficient for human language. The chimpanzee with language input shows that the human environment is necessary, but not sufficient for human language. What is sufficient is the combination of both: the human genome developing in the human social linguistic environment, with the specific interaction between the two producing the language system that is the defining cognitive achievement of our species. The children who were found after years of deprivation, who were studied and educated, and whose development was followed across years of remediation, have lives that did not follow the trajectory that the human developmental script specifies. Jeannie was eventually removed from the researcher who had been working with her, placed in a series of foster homes where she was reportedly mistreated and eventually lost to follow-up by the scientific community that had been tracking her development. Victor, the wild boy of Aveyron, spent his later years in the care of Itard's housekeeper, having plateaued in his development at a level far below what would be expected for a person of his age and apparent intelligence. Their cases are not simply scientific data. They are lives shaped by circumstances that were not of their choosing and whose consequences followed them permanently. The scientists who studied them did so in the context of a genuine care for the individuals involved, even when the research interests and the welfare interests of the subjects were not always perfectly aligned. The cases are important for what they reveal about the relationship between genome and environment. They are also important for what they reveal about the ethical dimensions of science, conducted with vulnerable individuals who cannot fully consent to their participation. The chimpanzees who participated in the language research are in a similar position. Individuals whose circumstances, whose life in captivity, and whose exposure to a human communicative system were not of their choosing, and whose welfare was not always adequately considered in the design of the research programs. Washoe and Nim and the other chimpanzees who participated in the language research were taken from their wild homes as infants, raised in artificial environments, exposed to a communicative system that was not their natural one, and in some cases transferred between research programs in ways that disrupted the social bonds they had formed. The cognitive sophistication that the research documented is inseparable from the specific circumstances that allowed it to be documented. And those circumstances raise the same ethical questions that the human deprivation cases raise from a different direction. What the comparison between the two experiments ultimately illuminates is not simply a scientific question about the relationship between genome and environment in language development. It is a philosophical question about what it means to be a member of a species with a specific genetic endowment, developing in a specific social environment, in the specific historical moment and cultural context that the accident of birth has placed you in. The human child is not simply the expression of its genome. It is the expression of its genome in the specific environment that the human community provides, and the community is as essential to the production of a full human being as the genome. The chimpanzee is not simply the expression of its genome either. It is the expression of its genome in whatever environment it develops in, and that environment shapes what the genome produces in ways that the experiments this video has described have shown to be substantial. Both species are the products of the interaction between their genetic inheritance and their developmental environment. And the experiments that have varied these conditions have revealed the interaction more clearly than any other evidence available. Tonight, in a world that contains both chimpanzees and humans, both species are developing in the specific environments that their circumstances have placed them in. The chimpanzee infant in the forest is learning the communicative system of its community through the observation and the social interaction that the community provides. The human infant in a language-rich home is acquiring the linguistic structures that the critical period is making available to it, building the neural architecture of language from the interaction between the inherited potential and the social input that surrounds it. The outcomes of both developmental processes are specific to the specific genomes and the specific environments involved, and the distance between those outcomes, while substantial, is less absolute than the simple species comparison would suggest. Both are cognitive systems in development, shaped by the interaction of inheritance and environment, producing minds that are adapted to the specific social and ecological worlds they inhabit. The experiment that this video asked about has been partially run in multiple forms across the history of the science. The results are complex, specific, and worth understanding. Sleep well. The specific research on the development of social cognition in children raised in conditions of deprivation, which extends beyond the language impairments that the Jeannie case documented, to include the broader social and cognitive consequences of early deprivation, has produced findings that illuminate the relationship between the human social environment and the development of the full human cognitive repertoire. The Romanian orphanage studies, which examined the development of children raised in institutional settings with severely impoverished social and cognitive environments in the 1980s and 1990s, found that early deprivation produced lasting impairments not only in language but in executive function, social cognition, and the specific neural architecture of the attachment system that underlies normal social and emotional development. Children adopted from Romanian orphanages into normal family environments showed partial recovery of many cognitive functions, with the degree of recovery tracking the age of adoption and the quality of the subsequent environment in ways consistent with the critical period account. The language impairments were among the most persistent, but the broader picture was one of pervasive effects of early deprivation on the developing cognitive system that extended well beyond the specific domain of language. The attachment research that underlies the understanding of the social deprivation effects has its own comparative dimension that is relevant to the question this video poses. John Bowlby's attachment theory, which identified the early caregiver relationship as the developmental foundation of social and emotional functioning, was developed partly from observations of children raised in institutional settings and partly from the ethological research on imprinting and attachment in non-human animals, including the famous experiments of Harry Harlow with rhesus macaques. Harlow's experiments, which raised infant macaques with wire or cloth surrogate mothers and documented the specific developmental consequences of the absence of a real social caregiver, showed that the primate developmental system, across species, requires the specific input of a responsive social caregiver for normal development to proceed. The chimpanzee infant and the human infant share this developmental dependency, and the effects of its disruption are qualitatively similar across the two species in ways that reflect the shared evolutionary history of the primate attachment system. The specific case of Genie, which has been the most studied of the language deprivation cases in the modern scientific literature, deserves the more detailed treatment that its complexity warrants. Genie was discovered in November 1970, when her mother took her to a social services office seeking assistance. The family had been living in isolation, the result of the father's paranoid belief that the world was dangerous and that his children needed to be protected from it. Genie's older brother had died in infancy, and Genie herself had been isolated in a small room since approximately 20 months of age, when her father decided that she was mentally retarded. The discovery of her situation triggered an immediate response from the scientific and clinical community, and within months, she had become the subject of an intensive research effort that combined clinical rehabilitation with scientific study. The combination of clinical and scientific interests in her case produced ethical tensions that have been extensively discussed in the subsequent literature. Tensions between the imperative to provide the best possible care and the scientific interest in maintaining the conditions that allowed her developmental progress to be documented as cleanly as possible. The trajectory of Genie's language development in the years following her rescue followed the specific pattern that the critical period account predicts with a precision that has made her case one of the most cited in the developmental linguistics literature. Her vocabulary development was rapid and showed genuine learning, with new words being acquired at rates that reflected real cognitive engagement rather than rote memorization. The specific words she acquired reflected her specific interests and her specific experience, showing that the vocabulary learning was shaped by her own cognitive and emotional world rather than by simple conditioning to the words that were most frequently presented in her environment. But her syntax remained severely impaired. The rule-governed combination of words into sentences with specific grammatical relationships, which human children acquire in the first 3 years of life, and which represents the most distinctive and most productive component of human language, did not develop in Genie in the normal form. Her utterances remained primarily content word strings, whose meaning depended on the individual meanings of the words and the context of their production, rather than on the grammatical structure of the combination. The neuroimaging research on Genie, conducted in the late 1970s, produced a finding that illuminated the specific neural consequences of her language deprivation in a way that connected the behavioral data to the underlying neuroscience. The language processing in normal right-handed adults is lateralized primarily to the left hemisphere, with the right hemisphere playing a secondary role. Jeannie showed an unusual pattern of language lateralization, with her language processing appearing to be primarily right hemisphere, the reverse of the normal pattern. The interpretation that was offered at the time, and that has been extended by subsequent research on language lateralization in deprivation cases, was that the absence of normal language input during the critical period had altered the normal developmental trajectory of language lateralization, with the right hemisphere developing the language processing function that the left hemisphere normally acquires during the critical period. The neural architecture of her language system was different from the normal human language architecture in a specific and measurable way, and the difference was consistent with the hypothesis that the critical period's role is to guide the development of the specific neural organization that supports normal language processing. The question of what Jeannie's experience was like from the inside, which is the question that the scientific and clinical records can approach but cannot fully answer, is one that the people who worked closely with her during the period of her rehabilitation describe with a specific combination of scientific precision and personal feeling that reflects the genuine complexity of the relationship between researcher and subject that her case produced. The linguist Susan Curtis, who was Jeannie's primary researcher and who documented her linguistic development in the most detailed analysis available, has described the experience of working with Jeannie as one of the most affecting of her professional life, one that produced genuine personal connection alongside the scientific documentation. The specific quality of Jeannie's cognitive engagement, the intelligence and the curiosity and the humor that emerged as she gained more ability to communicate, were visible to the researchers who worked with her and are described in their accounts with a warmth that the clinical language of the research publications does not fully convey. The broader history of cases of human development outside the normal human social environment, which extends across several centuries of documented reports and which has attracted the attention of philosophers and scientists since the Enlightenment, reflects a persistent human fascination with the question of what is natural and what is cultural in the human mind. The question of the noble savage, which Rousseau posed in the 18th century, and which the wild children cases were invoked to address by the scientists and philosophers of his era, is a question about the relationship between the human genetic endowment and the social environment that allows it to be expressed. The specific cases that have been documented across this history do not support either the Rousseauian picture of the naturally pure human mind corrupted by civilization or the Hobbesian picture of the naturally brutish mind civilized by culture. They support something more specific and more interesting, a picture of a genetic endowment for specific cognitive capabilities that requires specific environmental inputs to be expressed, and that is impaired in ways that are specific to the specific inputs that are absent when the normal developmental environment is not provided. The chimpanzee side of the comparison this video has drawn, which has been addressed in other videos in this series, and which the language research video describes in more detail, is relevant here specifically in the comparative developmental frame that the wild children cases open. The chimpanzee developing in a human social environment shows enhancements in communicative capability that the species' typical environment does not produce. The human child developing in a social environment that lacks the specific inputs of the human language environment shows impairments in linguistic capability that the species' typical environment would not produce. Both patterns reflect the same fundamental principle. The mind is not the genome. It is the genome in an environment. And the specific environment in which the genome develops shapes the specific mind that results, in ways that are bounded by the specific potential that the genome makes available, but that are not determined by the genome alone. The research on the critical period for language acquisition, which the Jeannie case and the other deprivation cases have contributed to alongside the experimental research on language acquisition in typical development, has produced a much more detailed account of the specific nature of the critical period and its neural basis than was available when the concept was first proposed by Eric Lenneberg in 1967. The critical period is not a single window that opens and closes at a fixed time. It is a series of overlapping sensitive periods for different components of language, each with its own developmental trajectory and its own relationship to the neural plasticity that underlies it. The sensitive period for phonological acquisition, the learning of the sound system of the language, closes earliest. The sensitive period for morphological and syntactic acquisition, the acquisition of the rule system that governs word structure and sentence structure, closes later. And the sensitive period for semantic acquisition, the learning of word meanings, remains open the longest and shows the least severe impairment in cases of late language acquisition. The specific pattern of Jeannie's language development, with her relatively preserved vocabulary development alongside her severely impaired syntactic development, is consistent with this account of the overlapping sensitive periods for different language components. The specific ways in which the deprived children's language differed from normal language, when their development was analyzed in the technical terms of linguistics, rather than simply in terms of the overall level of linguistic functioning, provide some of the most informative data available on the specific nature of the critical period. The analysis of Jeannie's language by Susan Curtis, which examined the specific structural properties of her utterances, rather than simply counting the words she produced, found that her language showed a specific structural profile that was consistent across her development, and that differed from the structural profile of normal language in specific and revealing ways. The absence of functional morphology, the grammatical markers that indicate tense, aspect, agreement, and case in languages that have them, was one of the most consistent features of her language. The limitation of sentence structure to relatively flat, non-hierarchical configurations, rather than the embedded, hierarchically structured sentences that normal adult language produces, was another. These specific structural features are consistent with the hypothesis that the critical period is specifically required for the acquisition of the rule systems that govern the syntactic and morphological structure of language, and that the semantic and pragmatic systems that support vocabulary acquisition are less dependent on the critical period. The comparison between the wild children's language and the language of chimpanzees and bonobos, trained in sign language or lexigram systems, reveals an unexpected parallel that is worth noting. The structural profile of Jeannie's language, with its relatively preserved vocabulary and its severely impaired syntax, resembles in some respects the structural profile of the language produced by chimpanzees and bonobos in the laboratory research. The trained great apes produce sign and lexigram combinations that convey meaning through the individual meanings of the symbols and through the specific pragmatic contexts of their production, rather than through the grammatical structure of the combination in the way that human language does. The wild children show a similar profile, with vocabulary available but grammar impaired. The parallel is not perfect, and the mechanisms that produce it are different in the two cases, but the similarity is informative. What the deprivation cases and the great ape language research together suggest is that the vocabulary component of language, the referential use of symbols to communicate about objects and events, has a lower threshold of environmental input requirement than the grammatical component, and that the grammatical component is the one that most specifically requires the normal human developmental environment during the critical period. The philosophical implications of the deprivation cases, which have been engaged with extensively in the philosophy of language and in the philosophy of mind, touch on some of the deepest questions about the relationship between language and thought. If language is the medium in which human thought is conducted, as some philosophical accounts of the relationship between language and cognition propose, then the absence of language in a child who has grown past the critical period without it should produce not only the absence of language but the absence of the specific kinds of thought that language makes possible. The Jeannie case provides some evidence for this view. Jeannie's cognitive performance on tasks that did not require language was substantially better than her performance on tasks that required linguistic mediation, suggesting that her cognitive capabilities in the non-linguistic domain were less impaired than her linguistic capabilities, but that the specific cognitive operations that language supports were impaired in ways that tracked her linguistic impairment. The question of what this means for the relationship between language and thought is one that the case illuminates but does not resolve. The specific question of whether the children found in animal environments, the most extreme version of the scenario this video poses, actually lived with animals, or were simply abandoned in wilderness areas where animal contact was incidental rather than constitutive of their developmental environment, has been actively debated in the historical and anthropological literature. The most credible cases, including the two wolf girls of Midnapore, whose case was documented by the Reverend J.A.L. Singh in 1926, and which has been the subject of extensive anthropological analysis, are not fully resolvable with the evidence available. The behaviors that Singh described in Kamala and Amala, the two girls he reported finding in a wolf den, including quadrupedal locomotion, nocturnal activity, howling, and the specific feeding behaviors of animals raised by non-human caretakers, are behaviors that could reflect either actual rearing by wolves or the specific behavioral adaptations of children with severe developmental disabilities who had been abandoned. The evidence does not allow definitive discrimination between the two possibilities, and the historical and anthropological literature has reached no consensus on the authenticity of the reported cases. What the wild children cases can establish, regardless of the specific circumstances that produced each case, is the general principle that the human developmental environment is required for normal human cognitive development to occur. The specific social, linguistic, and cultural inputs that the human community provides during the critical period of development are not optional enrichments added to a development that would proceed adequately without them. They are the developmental environment without which the specific human cognitive architecture does not develop in its species' typical form. This is true regardless of whether the absence of that environment is the result of neglect by human caretakers, isolation in a wilderness area, or the hypothetical scenario of literal rearing by a non-human species. The relevant variable is the presence or absence of the human social linguistic environment during the critical period, and the outcome of its absence is the same, regardless of what was present instead. The cognitive profile of the children who were found after extended periods of deprivation and who were subsequently given access to the normal human developmental environment shows a specific pattern of partial recovery that is itself informative about the nature of the critical period. The recoveries that occurred were most complete in the domains with the longest critical periods, specifically vocabulary acquisition and the pragmatic use of language in social contexts, and least complete in the domains with the earliest closing critical periods, specifically the phonological system and the syntactic rule system. This pattern of differential recovery across domains is consistent with the view that the critical period is not a single all-or-nothing developmental window, but a set of overlapping sensitive periods, each with its own developmental timing and its own relationship to the neural plasticity that underlies it. The partial recoveries that occurred demonstrate that neural plasticity for language does not end abruptly at the close of the critical period, but that the degree of plasticity that remains after the critical period is substantially reduced relative to what was available during it. The emotional and social development of the deprived children, alongside their linguistic development, reveals that the effects of deprivation extend beyond the specific domain of language to encompass the broader social and emotional cognitive systems that depend on the social input that the human developmental environment provides. Jeannie's attachment behavior, which was studied extensively by researchers who worked with her after her rescue, showed specific features that distinguished it from the normal attachment behavior of children who had experienced typical early social environments. She formed attachments to specific caregivers readily, but the quality of these attachments was different from normal child attachment in specific ways that reflected the disruption of the early attachment system that the deprivation had produced. The emotional intelligence and the social cognition that normal human development produces through the interaction between the genetic endowment and the social experience of the early years were developed in forms that reflected the specific nature of the environment in which Jeannie's early years had been spent. The research on the development of theory of mind in children raised in atypical environments, which has been conducted with children from Romanian orphanages and with children with autism and other developmental conditions that affect the normal social environment, has produced findings that parallel the language deprivation findings in revealing the environmental inputs that the normal development of theory of mind requires. Theory of mind, the ability to model the mental states of others and to use those models to predict and explain behavior, develops in a specific sequence in typical development that is influenced by the social experiences of the child across the first 4 years of life. Children raised in institutional settings with impoverished social environments show delays and specific impairments in theory of mind development that are consistent with the view that the normal social environment of childhood provides the specific inputs that the developing theory of mind system requires. The parallel with the language findings is instructive. Both language and theory of mind are capabilities that the human genome makes possible, but that require specific environmental inputs during specific developmental windows to be expressed in their species' typical forms. The question that this video has been building toward, which is not simply the sensational framing of the title, but the genuine scientific and philosophical question underneath it, is this: what is the human mind, and how much of it is in the genome versus in the environment? The deprivation cases and the chimpanzee language experiments together provide an empirical answer to this question that is more specific and more grounded than the theoretical accounts alone could provide. The human mind is the product of a specific genome developing in a specific environment, and both the genome and the environment are necessary for the species' typical outcome. Remove the environment, and the genome's potential is not expressed. Provide the environment to a genome that is different from the human genome in the specific relevant ways, as the chimpanzee language research did, and the environment produces enhancements without reaching the human level. The full human outcome requires both, and the specific way that the two interact is the subject that the research this video has described has been working to characterize for more than half a century. The children who were found after years of deprivation, whose lives were shaped by circumstances that no child should face, have contributed to the scientific understanding of the relationship between genome and environment in a way that was not of their choosing, and that came at a cost to them that the research literature acknowledges but cannot adequately compensate. Their cases are important. They are also individual human lives, and the individuals who lived them deserve to be seen as such, rather than simply as data points in a scientific argument. However important that argument is, the scientific knowledge that the cases contributed was produced at a cost that the individuals who produced it did not have the opportunity to consent to, and acknowledging this is part of what it means to take the science seriously, and to take the ethics of science seriously simultaneously. The chimpanzees whose participation in the language and social cognition research contributed to the understanding that this video has drawn on are in a similar position. They are individuals whose circumstances were not of their choosing, whose lives were shaped by the research programs they participated in, and whose contributions to scientific knowledge were made without the possibility of consent. The cognitive sophistication that the research documented makes this ethical dimension more rather than less significant, because the documentation of that sophistication is part of what makes the question of the animal's welfare more rather than less pressing. The research that reveals the intelligence of a being is research that simultaneously makes the ethical treatment of that being more urgent, and the chimpanzee research community has engaged with this tension in ways that have produced genuine improvement in the standards of care and in the ethical framework of the research, even if those improvements have not always kept pace with what the scientific knowledge about the animals involved would justify. Tonight, the chimpanzees in the research institutes and sanctuaries, and in the forests where the field research is conducted, are doing what chimpanzees do. The humans in the research programs that have produced the knowledge this video has drawn on are doing what scientists do, building on the work of the previous generation to produce the specific contributions that will shape the next generation's understanding. And somewhere in the world, there are children who are not receiving the human social linguistic environment that their developing minds require, whose potential is being constrained by circumstances that the scientific understanding of the critical period makes it possible to prevent. The knowledge that the deprivation cases and the chimpanzee language research together have produced is knowledge that has direct implications for the welfare of real children in real circumstances, and the translation of that knowledge into the policies and the practices that protect the developmental environments of children is one of the most important applications of developmental science available. Sleep well. The specific legacy of the Genie case, which extends beyond the specific scientific findings to include the broader ethical and social questions that the case raised about the responsibilities of the scientific community toward the vulnerable individuals whose circumstances make them involuntary contributors to scientific knowledge, is a legacy that continues to shape how developmental research is conducted and how the relationship between research and welfare is managed. The protocols for working with vulnerable research subjects that have been developed in the decades since the Genie case are partly a response to the specific ethical problems that her case identified. The broader framework of research ethics that now governs the conduct of developmental research with children has been shaped by the specific lessons of the cases where that framework was absent or inadequately applied. The children whose cases contributed to this framework did so at a cost that the framework is designed to prevent future children from bearing. That is not adequate compensation for what they experienced, but it is the specific form of legacy that science can provide, and it is worth acknowledging alongside the scientific knowledge that the cases produced. The final answer to the question this video posed, what would happen if a chimpanzee raised a human child, is an answer that the scientific research makes possible to give in specific terms. What would happen is this: the child's genetic endowment for language, for social cognition, for the specific human cognitive capabilities that the genome makes possible would not be expressed in the species' typical form because the social linguistic environment required for their expression during the critical period would not be provided. The child would develop as a human being in the biological sense that its genome specifies, but the cognitive architecture that the genome is designed to produce in the context of the normal human developmental environment would be shaped instead by the specific inputs that the chimpanzee social environment provides. The outcome would be a human being whose human potential was expressed in a form fundamentally different from the species' typical form, because the environment that the genome requires to produce the species' typical outcome was not the environment in which the development occurred. That is what the science reveals. And what the science reveals in this case is something that reaches beyond the specific question to illuminate the most fundamental fact about what it means to be human, that what we are is not simply in us. It is in us and in the world that shaped us. Both are necessary. Neither alone is sufficient. Sleep well. The specific neural mechanisms that underlie the critical period for language acquisition have been studied in increasing detail as neuroscientific methods have advanced. And the picture that has emerged connects the behavioral data from the deprivation cases to the underlying biology in ways that make the critical period concept more precise and more actionable than it was when it was first proposed. The critical period is associated with the maturation and eventual stabilization of the inhibitory neural circuits that regulate the plastic changes in the cortex during development. The opening of the critical period corresponds to the maturation of specific inhibitory interneurons that allow the rapid, experience-dependent modifications of cortical circuits that the critical period's enhanced plasticity requires. The closing of the critical period corresponds to the formation of structures called perineuronal nets around the mature inhibitory interneurons which stabilize the circuits and reduce their plasticity. This biological account of the critical period's mechanism connects the behavioral timing of language acquisition to the specific neural developmental events that produce the window and its closure, and it has opened the possibility of pharmacological and experiential interventions that might extend the critical period or reopen it after its natural closure. The research on second language acquisition, which examines the learning of a second language after the first language has been acquired in the context of the critical period, provides additional data on the nature of the critical period and its effects on language acquisition. Adults who learn a second language after the critical period has closed show specific impairments in the acquisition of the phonological system of the new language, producing the accents that mark non-native speakers, and show more variable but generally reduced acquisition of the morphological and syntactic system of the new language compared to children who learn the same second language during the critical period. The specific pattern of adult second language acquisition is consistent with the reduced plasticity of the post-critical period brain, and the comparison between child and adult second language acquisition is one of the clearest demonstrations available of the behavioral consequences of the critical period. The cases of deaf children who were not exposed to sign language during the critical period and who were subsequently given access to a sign language after the critical period had closed provide a specific and well-documented category of language deprivation case that has been studied in more controlled ways than the wild children cases. The language development of late-exposed deaf signers shows the same specific pattern as the verbal language development of the wild children. Relatively preserved vocabulary acquisition alongside impaired morphological and syntactic acquisition, with the specific structural features of the signed language showing the same kind of simplification and non-standard organization that characterizes the language of children like Jeannie, who were exposed to verbal language after the critical period. The consistency of this pattern across deaf sign language acquisition and hearing verbal language acquisition across very different cases and very different research methodologies provides strong support for the view that the critical period effects are specific to the linguistic structural systems rather than to the modality of language. The research on the development of home sign, which is the gestural communication system that deaf children spontaneously develop when they are not exposed to a conventional sign language, is one of the most striking demonstrations available of the human genetic endowment for language and its expression even in the absence of a conventional linguistic model. Deaf children in hearing families who receive no signed language input and who are not successfully learning the spoken language of their hearing community spontaneously develop a gestural system that they use to communicate with the hearing people around them. This home sign system, which is created by the individual child rather than learned from a community of signers, shows a number of the structural properties of conventional languages, including the consistent ordering of semantic roles within the gestural sequence and the use of specific hand shapes that function as morphological markers. The spontaneous development of these linguistic properties, in the absence of any conventional linguistic model, is the strongest available evidence that the human genetic endowment for language includes not just the capacity to learn a language that is provided in the environment, but the capacity to create a language when the environment does not provide one. The home sign research by Susan Goldin Meadow and her colleagues, which has followed specific deaf home signers across years of development and has analyzed the specific structural properties of their gestural systems in detail, provides some of the clearest evidence available for the specific linguistic properties that the human genetic endowment generates in the absence of a conventional linguistic model. The home signers produce consistent predicate argument structure in their gestural sequences, organize their gestures according to semantic role in ways that parallel the grammatical organization of conventional languages, and develop inflectional systems that track specific semantic dimensions of the events they are describing. These properties emerge without instruction, without a conventional model to learn from, and without the social community of signers that conventional sign languages are transmitted through. They emerge because the human genetic endowment for language includes the specific capacities that generate them, and because the developmental environment of the home signing child, while lacking a conventional language, provides the social communicative context that allows those capacities to be expressed. The comparison between the home signing deaf child, who creates linguistic structure in the absence of a conventional model, and the chimpanzee who acquires a limited version of a conventional symbolic system through training and social exposure, is one of the most informative comparisons that the research on language and its developmental basis has produced. The home signing child generates linguistic structure spontaneously. The trained chimpanzee acquires a version of it through extended exposure and training. The first demonstrates the generative, spontaneous nature of the human language endowment. The second demonstrates the extent to which the chimpanzee's communicative learning system can acquire elements of a linguistic system when the appropriate input is provided. Together, They illuminate the specific nature of the difference between the human and the chimpanzee cognitive architecture for language. The human architecture generates linguistic structure. The chimpanzee architecture can acquire elements of it. The difference between generating and acquiring in this context is the difference between a system specifically adapted to produce language and a system that can learn elements of language when provided with the appropriate environmental input. The story that this video has told, from Genie's rescue in 1970 through the Washoe research of the 1960s and 1970s and the Kanzi research of the 1980s and beyond, is a story about what happens when the normal relationship between the human genome and the human developmental environment is disrupted in specific directions. The disruption of the human genome's development in the human environment, which the deprivation cases document, reveals what the human environment contributes to the development of the human mind. The provision of a human-like environment to a non-human genome, which the chimpanzee language research represents, reveals what the genome contributes independently of the environment. Together, the two disruptions illuminate the relationship between the two contributors in a way that neither alone could accomplish. The science that the two research programs have produced is science that is more than the sum of its individual findings. It is science that has changed how we understand what we are. Not the simple expression of a genome, not the simple product of an environment, but the specific, irreplaceable outcome of the interaction between a particular kind of genome and a particular kind of environment. An interaction that produces the mind we inhabit, and that has been running in the same essential form for as long as human communities have been raising human children. Sleep well. The children who missed the critical period for language, and the chimpanzees who were given access to a version of the human communicative environment, have both contributed something permanent to the scientific understanding of the relationship between genome and environment in the development of the human mind. The contribution was not made by choice in either case. It was made by circumstance, in the specific circumstances that the research programs and the life histories of the individuals involved created. What science does with such contributions is to acknowledge them, to build on them as carefully and as honestly as the evidence allows, and to apply the knowledge they have generated to the welfare of the individuals and communities whose situations the knowledge is most directly relevant to. The knowledge is real. The cost at which it was produced is also real. Both belong to the full account of what the science has been and what it has done. Sleep well. The specific research on sign language genesis, which examines what happens when a new sign language is created by a community of deaf individuals who share no prior conventional sign language, provides another window into the human genetic endowment for language and its expression in novel social contexts. The case of Nicaraguan Sign Language, which was created by the first generation of deaf students to attend the newly established schools for the deaf in Nicaragua in the 1970s and 1980s, is the most extensively documented case of a new language being created from scratch by a community of language learners who shared no prior conventional linguistic model. The first generation of signers produced a relatively informal and highly variable gestural system. The second generation of children who learned this system as their primary language and who were exposed to it during the critical period for language acquisition produced a more structured and more systematic sign language that showed the specific grammatical properties that the first generation system had not fully developed. The creation of grammatical structure by the second generation of Nicaraguan signers from the less grammatical input provided by the first generation is the most direct demonstration available of the human language acquisition system's tendency to impose grammatical structure on the communicative input it receives during the critical period, rather than simply learning the structure that the input provides. The human child who is given a linguistic environment during the critical period does not simply learn the language that the environment provides. The child imposes structure on the linguistic input, building a grammatical system from the evidence the input contains in ways that go beyond what the input explicitly teaches. This imposition of grammatical structure, which produces the grammatically regular language of native speakers, from the variable and sometimes irregular input that the language environment provides, is one of the most distinctive properties of the human language acquisition system, and one that has no clear parallel in the communicative learning that the chimpanzee and bonobo language research has documented. The human child is not learning language. The human child is building language from input, using a cognitive system that is specifically designed to extract grammatical structure from linguistic experience. That is what the critical period is for. That is what the deprivation cases reveal when its inputs are absent, and that is what the chimpanzee language experiments reveal when the inputs are provided to a cognitive system that is related to the humans, but that lacks the specific language-building architecture that the critical period serves. Sleep well. The full picture of what this video has tried to convey is a picture of two experiments running simultaneously on different sides of the human-chimpanzee comparison, each illuminating a different dimension of the relationship between the human genetic endowment and the human developmental environment. The deprivation cases run the experiment of removing the human environment from the human genome, and they show what the genome alone produces, a mind with the potential for language and social cognition that the species' typical genome carries, expressed in a form that is impaired relative to the species' typical outcome because the critical period inputs were absent. The chimpanzee language experiments run the experiment of providing a version of the human environment to a different genome, and they show what the human environment alone produces in a non-human genome. Enhancements in communicative and social cognitive capability that move the chimpanzee toward the human outcome without reaching it, because the genome that the human environment is interacting with is different from the human genome in the specific ways that matter for language. Together, the two experiments triangulate the relationship between genome and environment in the production of the human mind. And what they reveal is that the mind is neither genome nor environment alone. It is what happens when a specific genome meets a specific environment during a specific developmental window. That is what this video has been about, and it is perhaps the most important thing that the science of human development has produced in the past 60 years. Sleep well. The child who is sleeping tonight in a home where language surrounds it, where the voices of caregivers provide the specific input that the critical period is processing and incorporating into the developing language system, is building something that the genome made possible and that the environment is making real. The chimpanzee in the forest or the research facility, whose communicative system was shaped by the specific social and ecological inputs of its developmental environment, is also building something that the genome made possible and that the environment is making real. The two outcomes are different. The processes that produce them are related. The science that has worked to understand both, across 60 years of research that has spanned both species and both directions of the genome-environment interaction, has produced a picture of the relationship between biology and culture, between inheritance and experience, between what we are born with and what the world makes of it, that is more specific and more grounded and more honest than any account that preceded it. This channel will continue to build on that picture, one video at a time. Sleep well. The question this video asked, what would happen if a chimpanzee raised a human child, was never primarily a question about chimpanzees. It was a question about what a human child is and what it needs to become, what its genome makes it capable of becoming. The answer, built from the cases of children who did not receive what they needed and from the chimpanzees who received a version of what humans receive, is a specific and grounded one. The human child needs the human environment, specifically the human language environment during the critical developmental window, to develop in the species' typical form. Without it, the genome's potential is not expressed. With it, even a different genome moves in the direction of the human outcome. Both findings are important. Both are the products of science conducted at a cost that the individuals involved did not choose to pay. Both belong to the record of what the research has produced and what it has cost, and both deserve to be acknowledged as such. Sleep well. The science continues, the question deepens. The children and the chimpanzees whose cases have contributed to the answer remain each in their own circumstances, living the lives that the science has tried to understand and that the understanding has not yet adequately